us on the call. My name is Manuel Chodibonji. I'm from Nigeria. Yeah. So I want to welcome each and every one of you to yet another webinar hosted by the Phyla Biotech. Without wasting much time, just to appraise us of the housekeeping rules. So if you have any question, please don't unmute your mic except you are called upon to do so. You can make use of the chat section if you have any questions in any of at the end of the sessions. There will be a question and answer. So at that time, they'll tend to your question. So in order for this to flow well and for us to maximize this opportunity, we need to please abide by that. Without wasting much time, I will call on Dr. Yusuf Agabi, who is a senior lecturer at the University of Jos and the director of Phyla Biotech, for him to introduce the theme of this webinar. Thank you very much. Over to you, Dr. Yusuf.
You are muted.
Dr. Yusuf, I think your mic is muted. Okay. For that, the people who will be speaking to us today are seasoned scientists in the field, and yes, their research is not something that's new to us. They are mentors and fathers in the field. The person of Dr. Simon Agwale, who will be our keynote speaker, Dr. Rodrick, Katete, who will be taking us on proteomics, data analysis, and lastly, Dr. Wadzani, Dauda, who will be taking us on a hands-on session for phylogenetic analysis. So, over to you, Dr. Yusuf Agabi, to introduce of the webinar briefly, then we can now move on to the...
Okay, thank you very much. Thank you very much, Benjamin, for that brief opening remarks and introduction. Dr. Kufri Joseph from Germany to kindly read the citation of our keynote speaker, Dr. Simon Agwale.
Thank you, sir. Without much ado, we already welcome our keynote speaker, and my work is just to read a citation. Dr. Simon Magaji Agwale is a renowned virologist and vaccinologist. He has dedicated his 25-year career to developing novel vaccines and leading large-scale projects. His expertise spans from area of vaccine development, molecular epidemiology, and research management. Dr. Agwale is the founder and the CEO of Innovative Biotech Limited in Nigeria and Innovative Biotech in the US. His scientific leadership has been instrumental in shaping the future direction of vaccine development worldwide. That is very, very important. And we know that from this other side of the world, Africa, we are always up. Dr. Agwale has developed several candidate novel vaccines utilizing BLP vaccine platform and licensed several others, including vaccines for Ebola, HIV, typhoid fever, Lassa, and yellow fever, as well as COVID-19. He has secured some funding from both the African Export-Import Bank and International Trade Center, Geneva, to build the first Africa's modern end-to-end vaccine manufacturing facility in Nigeria. This is very incredible and commendable. His research expertise spans various reputable institutions in Nigeria. He's been in the University of Jos, Brazil, Germany, in Robert Koch Institute, very important institute here in Northwest Germany. He also worked in UK, Imperial College, and the US, and many other places. He has represented Nigeria and Africa at various international scientific fora, including World Health Organization and European and developing countries clinical trials partnership, EDCTP, as well as Africa CDC, Africa Vaccine Manufacturing Initiative, and lots more. Dr. Agwale's expertise has been recognized globally with appointments to prestigious organizations such as EDCTP, Vice Chair of African Vaccines Manufacturing Initiative, Board of Directors, and lead and member of Africa CDC AVM's Technology Transfer and IP Bold Program and Talent Development work streams, respectively. Throughout his career, he received several scholarships, fellowships, and grants from reputable organizations. He has authored and co-authored several publications in reputable scientific journals of high impact. His contributions to the field have been recognized globally, and he continues to strive for innovative solutions to combat infectious diseases. So finally, our speaker has traveled widely to represent Nigeria and Africa in many countries of the world, even I would say widely from Nigeria to everywhere in Switzerland, Belgium, Sweden, Norway. So it's not just a Nigerian, he's not just an African, but it's a global person. And I know for the importance of vaccine development and vaccine administration and all that, he is a very important person to tell us more and about what will make us be able to vaccine. So thank you, sir, for coming on board, and we give you the time and to go ahead.
Well, thank you very much, organizers, for asking me to give this keynote address. I'm going to be very brief and precise. My job basically is to talk about how we can use genomics and proteomics to develop vaccines and then set up a vaccine manufacturing facility. The other two speakers will tell you more about proteomics and genomics, but basically I'm going to focus on translation, how we can use genomics and proteomics to develop vaccines and then to set up vaccine manufacturing facility from a practical point of view, because that's basically what I've been doing for the last 30 years. So what's going on? I'm not able to move to the next slide.
So can you stop sharing and reshare again?
Starting. Okay. Okay. Okay, briefly, I will like to define genomics, proteomics. As we all know, of course, genomics is the study of DNA, genes, and genetic variations. And the so-called genomics era significantly shifted the paradigm of vaccine development from just the microbiological to sequence-based approaches. A typical example is the SARS-CoV-2 genome sequencing, which I'll talk to you briefly, and then the HPV genotypes in Africa. As you know, the HPV vaccine today is based on predominant genotypes. So that's why it is important to continue to sequence the viruses in circulation because, I mean, one of the recent data clearly showed that the current HPV vaccine, even the HPV-9 vaccine, does not protect against HPV 35, which is only circulating predominantly in Africa. So there's a need to update the current vaccines that are being used to include HPV 35. So I'm just saying this for us to see the importance of genomics in vaccine development. So it has been exploited to perfect the identification of targets, to design new vaccines, and to predict their effects in patients. Again, proteomics, as you all know, is the study of structure and function of proteins. And it enables the identification of antigens and epitopes, as we're going to see, example of the spike protein characterization for COVID-19 vaccines, the famous Yuan strain. And you know that all the early COVID vaccines were based on the Wuhan strain. And that's the importance of genomics, because without that spike protein from Wuhan strain being sequenced, it will have been difficult to rapidly develop and deploy COVID vaccines. So the next question is, what is a vaccine? A vaccine basically is a biological preparation that, when inoculated or administered, induces two arms of immune responses. We are not going to, I mean, this presentation, like I say, is practical translational, but there's different mechanisms of how to induce antibodies. The antibodies are supposed to protect against the pathogen. And then you have the cellular immune response. Should the antigen not be able to induce sufficient immune responses, you have the cellular immune response. Even after infection, the cellular immune response will be able to kill infected cells. So the goal is to protect against the pathogen or suppress chronic infections. So as we all know, vaccines are presently the most powerful and cost-effective health interventions. I give you an example of typhoid vaccine, where we have a conjugate vaccine that potentially protects for life for a fraction of what you need to treat. If you look at the the strains circulating in Africa, they are resistant to all the antibiotics that are in use today. And the only antibiotic that treats current typhoid multi-drug resistant costs about $2,000. And as you all know, this is highly prohibitive for people living in Africa. So that's why it is important to take vaccines, low cost, to protect against diseases. They've been successful globally in the eradication of transmissible diseases, the smallpox, polio, measles. As we know, there's the emergence of measles today. Most of the kids, especially here in the US, that are infected are people that didn't take the vaccine. So that shows you the effectiveness of vaccines, and then the cost effectiveness of vaccines. Smallpox was eradicated because of vaccines. We're talking about polio eradication today because of vaccines. So this is basically what happens if one is not vaccinated. If you're not vaccinated, you have not induced the two arms of immune responses. You don't have the antibodies and the cellular response, and therefore the pathogen will overwhelm the immune system. But before your exposure to the pathogen, if you are vaccinated, this is what happens. You already have the antibodies, the cellular immune responses to suppress the pathogen. So the next question is, how are vaccines developed? The roadmap to vaccine development. This is an ideal situation because for us as biotech companies operating on the continent of Africa, we have to do everything because our universities are not doing any of these things. So we get involved in identifying the natural mechanisms of protection, which is called the correlates of protection. So what protects? If there's an outbreak, some people are infected, some are not. Some people die, some don't. So why are these other people not infected? Why are they not dying from disease? What are the correlates of protection? What correlates to the protection? So this job is supposed to be done by universities. Unfortunately, we're not living up to our tasks on the continent of Africa, except for South Africa, that's doing high quality, especially at University of Cape Town, high quality research to provide the data to the industry to take it to the next level. And that's how the entire ecosystem is set up. So once you know the correlates of protection, what protects, then you isolate the antigen that is responsible for the protection. And this is where genomics comes into play. What are the antigens that are responsible for the protection? So you isolate those antigens, you sequence them, that's genomics. And then you cannot design your vaccines, test them in animal models to see and challenge those animals to see if the antibody, against this targeted this antigen protects against the infection. So the next step is, okay, we know the antigen that is responsible for protection. We know that this antigen protects against the pathogen. And then the next question is, how can we present this antigen? So that's why you hear different vaccine platforms. The viral vector platform, which came into focus during the COVID pandemic. You have the DNA, you have the RNA, you have the inactivated, you have the protein subunits, and so on. So these are platforms to present that antigen that protects against the infection. So when the academics does this, the academic patents the technology, and then the industry, companies like us and other industries, we now go to the universities and say, oh, okay, you have this patent. So how can we license this patent and then be mutually beneficial? It comes at a cost, because you did all the research and everything, and you have the patent. And then the industry licenses the patent and commercializes it and pay royalties to the university. So the industry will now pick what the university did. And then our job is, of course, they did everything under research environment. The job of the industry is, okay, how can we scale this up? Because you use maybe 5-liter fermenters in the lab. You don't know the number of doses that the 5 fermenter will give. So the industry will now figure that out, which is called process optimization and scale up. And then the industry does the safety to show that the vaccine is safe in animal models, which is called the preclinical testing. And then the industry now will produce a clinical trial lot. That's good. The GMP is good manufacturing practice because all the academic did was non-GMP because their product is not supposed to go to humans. They are just making the vaccines, testing in animal models, and so on. And then the industry, now the vaccine is going into humans. How can we take it now? Do the safety, make sure that it is safe before we produce the clinical materials for clinical trials. So this is the roadmap for vaccine development, clearly showing the role of the our academic institutions, and then the industry. So once this is done, you've shown in animal model that the vaccine protects. You've shown that it is safe. Then you now move to clinical trials. The data generated or you got during preclinical testing will it be repeated in humans? So that's why in phase 1 trials, you recruit few volunteers because this is it's a new product. You want to see whether it is safe first in humans. So you can't expose thousands of people to a product. Of course, you know that it's safe in animals. You start small, 10, 20, 30, 60 volunteers, depending on the now move to phase 2, increase the number of people. So you now look at the safety and the effectiveness of the vaccine. And once those are established, then you go into large-scale clinical trials involving thousands of people to establish the efficacy of the vaccine in humans. And then once you show that this is the traditional ways of doing that, so once you show that the product, the vaccine is effective, then you now build a vaccine facility because it is during this for the manufacturing of the vaccine, and the vaccine facility is designed based on the process. And the reason we got COVID vaccine early within a year was because of government intervention. So the government say, okay, you don't need to worry about whether the vaccine will work or not. But the commercial organization companies will want to see that the vaccines work first before they build factory. So the US government said, oh, okay, we'll pay you produce the vaccine in advance. If the vaccine does not work, we lose as government, not the company. So that encouraged the companies to go ahead because the government has taken the risk. So I don't want to, because of time, go into details, but this is what happens in proteomics, genomics. You can see the traditional vaccines, inactivated vaccines, war virus vaccines fall into this category. So if you give the vaccine, this is what happens within the cytoplasm of the cell. They translated into proteins and then eventually into peptides. And the peptides are now, of course, I won't go into the detailed immunology. This is not the forum for that. And then the immune system recognizes those epitopes and then make specific antibodies to protect against future exposure. Then you have the second category of vaccines, which is the protein-based vaccine. These ones, you've made the protein, using different, whether mammalian cell, yeast, and so on. So you make the protein. So you are just injecting the protein. And this is where the VLP vaccines, the protein-based vaccines category fall into. And then because you are just injecting the protein, everything ends within the cytoplasm. The proteins are broken into peptides or epitopes, and then they are picked up by the immune system, and then antibodies are induced. But if you look at the newer vaccines, the viral vector vaccines, the nucleic acid vaccines, you can see that you're injecting the DNA. And in this case, the viral vectors will just deliver the DNA and it goes straight into the nucleus. So you can see the traditional vaccines has no interaction with the nucleus. Everything else within the cytoplasm because you make your protein outside the cell. But in this case, you are making your protein using the human system to make the proteins. So you inject the DNA and the DNA goes into the nucleus, transcribed into RNA, and then you have your messenger RNA produced, and then the messenger RNA comes back to exported to the nucleus, and then they are translated into proteins and then broken down into epitopes and then recognized by the immune system. Then if you look at the mRNA, that is for DNA. So you make the mRNA, the RNA here, and it's exported to the cytoplasm. But if you give mRNA, which was the main vaccine during COVID, again, it's mRNA, you have made it outside the cell, unlike the DNA that has to be transcribed to RNA, translated into proteins and then epitopes, and then recognized by the immune system. So how did we use this knowledge to advance development of our vaccines and then working towards setting up a vaccine manufacturing facility? So to make an HIV vaccine for Nigeria and other countries that, also having the same strain, subtype G and then recombinant AG, like Nigeria. So the first thing to do was to take samples from the entire country. This is the first and probably only study that look at molecular epidemiology of HIV in a single country, in a single study. So we clearly show, as you can see, that the predominant subtype at that time were AG recombinant and G. And then this, you can pick this article, look at the details, published in 2002 in Vaccines. So you can see here now, of course, the focus is to prevent infection. So you have to look at the envelope antigen. And this is the sequence that we use to make the vaccine. This is the amino acid sequence, that we use. So you can't change the amino acid sequence, but you can change the nucleotide because it's shown that if a sequence is CG rich without changing the amino acid, it tends to be more immunogenic than sequences that are not CG rich. So we have to engineer the sequence to make them CPG. So you can see the entire sequence here is CPG to make it more immunogenic. So I won't go into the detail that. And then now you look at the platform that you now use to make the vaccine. In this case, we use the baculovirus system and then clone the GP120 and then the GAC protein expressed in the insect cell, and then you have your vaccine. And this vaccine now, you vaccinate, you test first using electron microscope, to see whether the VLPs are made. And then you do your Western blot to show that indeed you've been able to express your proteins of interest. And then based on that, you now immunize your animals, you take the antibodies and test for effectiveness in the case of HIV you're looking at neutralization. So you can see that this vaccine that we made neutralized not only the Nigerian virus, but you can see even subtype C circulating in Africa, in South Africa, the MN, subtype B in Europe, and so on. So when Ebola hit, we use the same platform. As you can see, the glycoprotein of Ebola virus, which is the outer coat, we use that using the platform, made Ebola vaccine, the VLP approach, and then express that, create a VLP vaccine against Ebola. And then, of course, you have to do, for VLP, you have to do your electron microscope to make sure that the VLPs are made, and then you do your Western blot to make sure that the proteins are expressed. And this data, you can see a single dose induce a very good immune response. If you boost, that is the second dose, you get very high antibodies that will protect against Ebola. And this was presented during the Targeted Ebola International Conference in Paris in 2015. Now, when COVID hit, again, you can see this is the Wuhan strain. So the good thing that the Chinese did was immediately to sequence the spike protein, and this is the sequence of the spike protein. So that sequence was available globally. So different companies, different universities, different institutions took that sequence and then started making different vaccines. This is the receptor binding domain, because if you make antibodies against the receptor binding domain, the virus will not enter the cell. So this is the power of genomics. Once this data, you don't need the virus. Once you have the genomic, you can sequence the whole thing and create, the vaccine without even having access to the virus. So everybody was focused on spike protein, but at that time we made a vaccine that is focused on all the structural proteins. Again, you can see from this presentation that you have your vaccine here expressing different gene proteins of the SARS-CoV-2, and then expressed in mammalian cells. So you have your COVID VLPs here. You have to do your electron microscopy, and then, of course, your Western blot vaccination. Everybody was looking at 2 doses, at that time. And then you can see here to show that the vaccine that we made was highly immunogenic, using different adjuvants, and then to test for neutralization to see whether the antibodies neutralize. And this, again, that time, we made a vaccine against beta variant, and then we showed that it also neutralized the Delta variant. So, of course, vaccine development is done in partnership. So at that time, we partnered with Millipore Sigma, which is the US arm of Merck, for the entire process. We wanted to do the clinical trials in South Africa. And then, of course, the infection went down and we couldn't proceed with the COVID vaccine. So, but the process was developed, and then the facility designed based on the process that was developed. So here you can see now this is the factory, and this is what we are building now with support from Afromics. We are now working towards moving, because the feasibility study is ongoing to build this factory. And then once that is done, the entire modular facility will be built in Europe, and you will ship it to Nigeria, and then start producing these vaccines, VIP, that are based on this process. One other interesting vaccine that we are working on, I won't go into details of genomics, preclinical, clinical testing, but this is a Lassa vaccine that shows 100% protection, both protected against the Josiah strain, which is from Sierra Leone, and then the Nigerian strain, 100%. In both guinea pigs and then non-human primates. So we have developed a process. So everything boils down to developing the process. So we have developed the process for manufacturing Lassa vaccine. So in our factory, in our plant factory, we are going to have the VLP arm to produce the vaccines that use the VLP technology, and then the virocell. This one produces virocell or yellow fever Lassa vaccines will be used to do this. So this is the partner US facility where we are producing the clinical material. We have the clinical materials for conducting phase 1 clinical trials of Lassa vaccine. It was produced in this lab. So in summary, genomics and proteomics provide a foundation for identifying disease targets and vaccine antigens, and translational research transforms sequencing and protein data into vaccine candidates. Modern vaccine manufacturing plants integrate R&D labs, GMP suites, quality control, quality assurance, and cold chain distribution. And building local capacity ensures rapid epidemic response, healthcare security, and economic resilience. And then strategic investment, partnership, and workshop development are essential for success. So again, like I said, these studies are not easy. You need the financial partners, you need the technical partners, the tech transfer partners, the regulatory partners, to make this a success. So it's not an easy road, but again, it's a worthwhile adventure. So I thank you very much for your time, and I'll be happy if you have questions. Thank you. Over.
Wow, thank you very, very much for that enriching session, Dr. Simon Agwale. It was really a powerful one, taking us through, from the vaccine manufacturing to to the manufacturing plant. It was really an enriching session. So we are glad to have you speak to us today, and to the participants of this webinar. If you have any questions, please do well to drop it in the chat section, so doctor can be able to attend to them.
I think it would be nice to take all the presentations so that we don't waste time, and then you can have
Okay. Yes, we are just trying to buy time so as to prep the next speaker. Yes. So
okay. Ben, I think you can introduce the next speaker so that we can make progress over time.
Okay. The next our next speaker will be Dr. Rodrick S. Katete, who will be taking us on proteomics data analysis. His bio reads, Dr. Rodrick Katete is a principal investigator in the Molecular Medicine Research Group and founder of Afromics, which is a startup medical biotechnology company. He teaches general and analytical biochemistry, clinical chemistry and toxicology, medical genetics, and molecular cell biology at Mzuzu University, Mzuzu, Malawi. He has previously taught at Levi Wadzani Medical University, Mukuba University, and Texila American University, Zambia. He holds a Ph.D. in biochemistry from Sheffield University, a BSc in biochemistry and cell biology, microbiology, and biotechnology, and a BSc with honors in biochemistry and cell biology from the University of Witwatersrand, Johannesburg, South Africa. He did his postdoctoral research fellowship at the University of Cape Town, South Africa. His current research interest is in structural Christology. He has supervised 3 MSc students at Levi Mwawasa University, Medical University, 1 Ph.D. student at Texalia American University, and mentored many MSc and PhD students. Over to you, sir. Thank you very much, sir, for availing yourself. We are glad to have you. Over to you, sir.
Thank you very much. And also, I'd like to thank Dr. Agwale for a wonderful presentation on vaccine development. And it's very wonderful that we are trying to develop vaccines in this continent. It's very, very important. We had very difficulties during the COVID. I think mostly we were begging across the globe to help us, from Chinese, Americans, Russians, British. We tried all over the world to help us under the pandemic. So if we will be able to develop these vaccines, it will be very, very wonderful. And going forward, it means we have a very good future. So thank you very much for that wonderful presentations. I really appreciate, and I also learned a lot. And we have also learned a lot from your wonderful and good profile, which I've seen. So there's a lot that myself, I'm sure I will learn from you. So the next talk is on bioinformatics for beginners. So there'll be basics on bioinformatics so that even those that have not done bioinformatics before, they should be able to appreciate the importance of bioinformatics in vaccines or even drug design and many, many other things. So we'll be going through that as we go, and we're going to show a basic example on how we use bioinformatics in novel antimicrobial drug targets and biochemical pathways. So we're going to show 2 papers which we are just publishing just now on how we are using these tools in drug discovery. So basically, we will look at the introduction, definitions, objectives of bioinformatics, tools that are used in bioinformatics, application of bioinformatics, and we emphasize our application more will be on the antimicrobial drug targets and biochemical pathway that will come across. So this is basically what we'll be going through. So bioinformatics is a scientific discipline that involves the analysis, computation analysis, storage of biological data, and the word bioinformatics is derived from 2 terms: bio, which is biology, and informatics, which is data processing. So most of the time in bioinformatics, we focus more on data processing, and it comes from the bioinformatics. So it's a field which biology, computer science, and information technology merge into a single discipline for managing and analyzing biological data using advanced computer techniques. So majority of the bioinformatics that we'll be talking about has to involve especially ourselves as biochemists, myself as biochemist, we're more interested in the retrieval, storage and retrieval of data. So if we have got information, let's say we have sequenced a DNA, we need to deposit that. Most of the time we deposit it in National Center of Bioinformatics Information and other bioinformatics websites, and then others will retrieve that information, or ourselves also retrieve other information deposited by other people. So overall, bioinformatics can be defined as a storage, analysis, and search, retrieval of data. So nucleic acids sequence from genes and mRNA, and amino acid sequence of structural information of proteins. So altogether, bioinformatics could be looked at mathematical, statistical analysis, biology should be involved, computer science should be involved. As these fields advance, bioinformatics also get more and more advanced. So these 3 fields are very important that they work together. The more technologies are developed in the molecular biology pathway, the more also we are going to be able to use bioinformatics to deposit that information. The more mathematical formulas are developed and helped to analyze data, statistical formulas, all these put together, they help in the development of bioinformatics. So what are objectives? What are objectives of bioinformatics? Data management, to organize and store enormous volume of biological data. So these biological data could be anything: DNA sequences, protein sequences, protein structures. There's protein data bank, which stores a lot of protein sequences, gene expression data, and many, many other data. Could be mutation data. All those information are stored and have to be managed. Tools, the certain tools that are developed so that they can help us to analyze this huge amount of data. So there are computational tools that are going to be looking at each of these as we go along. So we are going to have algorithms that are developed, statistical methods that are developed to analyze and interpret this biological data so that they make sense. So we are able to understand what is going on. For example, if we have got, as we have heard previously from COVID issue, I've got a new strain of COVID, we want to know what are the difference from the previous COVID, let's say Alpha or from Omicron, what are the difference in these strains, and all that should give us an idea, especially ourselves as biochemists, to be able to see if there's a change in the amino acids, what are those changes mean? If the change from, let's say, fluorine to glycine, what does that mean in terms of the structure and the function of that particular protein? Let's say it's a spike protein. So biological understanding to achieve deep understanding of fundamental biological processes, analyze complex database and vary patterns within them. So we are also going to we also use biological information. We are going to see one of our publications on the same on how we are trying to elucidate the biochemical pathway of one of the bacteria that is involved in read biomediations. We know that that bacteria can do the job, it can do read biomediation, but we don't know what proteins and what biochemical pathways are involved at each stage of that. So biological understanding is also involved through bioinformatics. We can be able to elucidate a lot of things that a particular bacteria can actually do by looking at the specific proteins that it has and comparing it with other proteins. Gene and protein analysis, identify genes and proteins. That is very common these days. Everything goes up to molecular level. We always say that everything goes to molecular level. If we have got a protein sequence, most of the time we want to analyze if there is a disease, any strain, anything that has come out, the first thing that we rush for is to sequence it. Once we sequence it, we want to analyze and understand what is going on. We can also use bioinformatics to analyze the evolution studies. Most evolution studies, evolution relationships of genes, proteins, organisms, all these things involves bioinformatics as well. So these are some of the objectives of bioinformatics. So system biology as well is also involves bioinformatics, analyze and catalog biological pathways and networks to understand how macromolecules interact with each other and how those interactions influence biological processes. So we can also use bioinformatics to help us understand how systems are organized and then they produce a specific metabolic pathway and understand that metabolic pathway. So these are some of the objectives of bioinformatics that we mostly use. So we'll go through some of the tools. That are used in bioinformatics. So bioinformatics tools are software programs, and some of them we can pay. Some of them are readily available for free, depending on what you want to achieve. So they help to extract meaningful information from mass of complex biological processes and informations. And from there, we can be able to understand what is going on. After information of database, all tools are available for search and sequence in a sequence database. So categories of biological tools. So we have got biological databases, homology similarity tools, protein function analysis tools, structure analysis tools, sequence manipulation tools, and sequence analysis tools. All these tools are used at different depending on what you want to achieve. And also, when I'll be going through some of these tools, does not mean that those that have put are the only ones. There are many out there. There are many tools that are available. For example, for biological data processing, there are so many other tools there that are available on the internet that you can use depending on what your objectives are, what you are actually targeting, what you really want. So biological tools that are available. So we have got these are databases containing genomic, proteomics, and metabolic database. The data includes nucleic sequence, genes, and amino acids. So some of the major biological tools are biological databases are nucleic sequence database, DNA variation database, gene expression database, microbial genome database, and organism-specific genome database and protein database. Some of these tools, like I said, databases can be found within one single website. So, for example, the National Center for NCBI, the National Center for Biotechnology Information, you may find that my nucleotide sequence database is there. DNA variation, it's there. Gene expression, sometimes microbial genome database, so it's there. So you can find a lot of protein databases, so it's there. So you can find that most of these tools can also be found within one single website. So depending on what you want to do, you can choose which database you want to use to get. If you are playing around with proteins, then you can say, okay, let me go after protein database. and within the protein database as well, we can have many other different, could be human, Homo sapien database, could be microbial database, or within microbial database, we can also have different organisms as well, different bacteria, and then you can say this is specific bacteria database, so that many of these, all these can be found within, for example, the NCBI. You can have all that. So depending on what you are trying to achieve, you can use a specific database. So within those databases, some of the things that we do homology and similarity tools. There, most of these homology 2 sequences are nucleic sequence and protein sequence that share a common evolutionary origin with other sequences, indicating shared ancestry. So they share common evolutionary origins. This could be orthodox, that descends from common ancestor, or could be paradox created by gene duplication event within the same genome. So all these terms are also very important depending on what you want to be doing. So if you want to make sure that you are designing a drug, for example, that shouldn't target other proteins, or you want to remove a lot of other proteins that are involved and should just target any other protein, then you can make sure that you remove as many paradox as possible. So you don't want gene duplication. You want essential proteins only. The proteins that are essential are important and are also not paralogous as much as possible. Then the paralogous ones as your target. So we do all that. So this set of tools can be used to identify similarities as well for novel query sequence, for unknown structure, so or a function. So you can have a sequence which you have no idea what its function is, what its structure is. We always say that the structure determines function. So once we know the structure, we can be pretty much more or less predict the functions. Just similar to what the DNA structure. Those days when they discovered the structure of the DNA, they were able to know how it's going to replicate even before the replication process itself was well known. So they looking at that structure, they were able to know what it can do. So the same way with proteins, once we know the structure, then we can be able to predict the function. So from unknown structure, which is never known, they can compare it to known structures as well, and then we can move on to know how we can do, how we can deal with it. So some of the tools as well that we use for alignments, alignment of 2 sequences or more is called BLAST, Basic Local Alignment H2, which is also found on NCBI. We've got Basic Local Alignment H2, finds legions of local similarities between sequences. Program compare nucleic or protein sequences to a sequence database and then calculate statistical significance of those. So then the more significant it is, the more likely that these 2 sequences are the same, then we can use them for whatever we want at that stage, whether to design a drug against it or something else. So BLAST can be used to infer functions or evolution relationship between sequences or identify members of the gene family. FAST format is the format which we normally use when we are working with BLAST programs. Because of time, I always welcome questions afterwards if you have on these. So some of the tools that are available, I say that these are not the only tools. There are many tools out there that do also sometimes similar functions, depending on what you want. So some of these tools are protein function analysis tools. We have got InterProScan. InterProScan provides functional analysis of proteins by classifying them into families, predicting domains, important sites of this particular protein. So I'll be referring here and there to the a paper which we just been accepted for publication. I'll be referring to that here and there. That, for example, some of these tools, how we have used them. So we can use these tools to predict important sites within the protein. So we can have the protein. We want to know which part of this protein is very, very important for its function. Most of the time, those regions tends to be conserved. They tend to be conserved, and are not mostly they are not going to be overly mutated. They can be mutated, but not overly mutated, because those are very important regions of it. So those are some of the areas that we can use to target that particular protein. So we can have also PP search, that will compare your query protein sequence against all patterns stored in ProSite pattern database and determine the functions and characterize proteins. I've got RADA, rapid automatic detection and alignment repeats, which checks the repeats, and other structure analysis tools. I've got LastMore, Prospects, Capia. All these are some of the tools that are used to analyze protein structures. Some of them, they are also used like the Rutherford and Rutherford 2. I think Rutherford 2, they also got a Nobel Prize on Rutherford 2 very recently. It's one of those tools that can be used to predict a 3D structure from the primary structure, from the amino acid sequence. We can use that to predict the 3D structure of the protein based on homology modeling. So because of that, it can help us to predict proteins structures that are unknown, that we don't know their structures at all, that we haven't solved their structures using known structural elucidation techniques like crystallography, X-ray crystallography, electron microscopy, cryo-EM, all those techniques that have been employed. So the other one is Swiss PDB viewer. It's one of those as well, that we can download the protein structure data, coded files. Once you download the protein coded file from protein data bank, for example, then you can use the Swiss PDB viewer to look at that structure in 3D. So you want to see the structure in 3D as much as possible, or sometimes see what happens when you remove a specific amino acid, to what the impact of that into the structure itself. So that is one of the it's freely available. All the I can say that most, in fact, all the ones I have been talking about, all these ones are free of charge. So you can download PDB viewer, for example, it's for free. Then you can use the protein PDB viewer after you have downloaded the protein coded data from protein data bank. Then you can use PDB viewer to play around with your structure, to view it and turn it around and see what happens if you are removing specific amino acids and what happens to that structure. And we'll also look at bond angles and all sorts of other things on it. Mainly, these are the ones that you'll be targeting if you are designing a drug. Bond angles, hydrogen bondings, and if you are going to bring a ligand to it, what is going to happen? All those things can be able to be viewed on PDB viewer. The other ones are sequence manipulation tools. So these are repeat masker. It is a program that screens the DNA for interspaced repeats. The WebCat is a tool that helps us in instructional analysis, silent mutations, and single nucleotide polymorphisms analysis. And we have got translate. This is just a tool as well, which you can, once you have sequenced the nucleotide, the DNA sequence, you can then use this to translate that into a protein. And from there, you can go all the way to use other tools like AlphaFold to decide the structure, to deduce the structure of that specific protein. I've got primer blast or primer design. Those have done PCR. So this also will help you to see the hairpins, primer dimers, temperatures, TM, and many other things that are involved in primers as we are designing primers. So these are some of the tools that can also be used to manipulate the sequence depending on what you are trying to achieve. So overall, bioinformatics is very important. It's used for many things. It can be used for drug design, can be used for molecular modeling, protein sequence analysis, DNA sequence, molecular dynamics. All these things are some of the applications of bioinformatics. And mostly for ourselves in this part of the world, for example, in Africa, and many even other parts of the world across the globe, bioinformatics help us very much to decide before we can go into the wet lab which proteins are very important and analyze them as much as we can. And before we can spend so much money buying all the equipment and doing all the research and find out that that particular protein is not the one that we are interested in. For example, if you have got a protein which you think you can target with a specific drug or a vaccine, with a specific vaccine, but you want to know, is this protein also available in human genome? And the human genome is very big, it's enormous. So in the proteomes of human, is this protein going to be available? How do you do that? We use bioinformatics tools. And through bioinformatics tools, we can be able to say, okay, this protein also can be found in humans, or this protein can be found in gut microbiome, which are very important. And so if we design this particular, we target this particular protein, we can also end up targeting human proteins, and that's not going to be good. So before we can go all that far, bioinformatics in this part of the world can help us to say these ones we don't include, these ones we include. So that's mainly what we do. So example of what we have been doing is we did this in Enterococcus faecalis, antibiotic resistance, broad spectrum drug targets identifications using subtractive genomics. It's a paper which has been accepted and it's going to be published soon. The idea here was to find novel proteins that this specific bacteria has, that for starters, this particular bacteria is resistant to all antibiotics. So all known antibiotics is resistant in one way or another. So that's not good. So the new idea now is to say we need to find new drug targets. Drug targets that the current drugs are not targeting, and these drug targets should be novel. They have never been discovered before this particular bacteria. So then we use bioinformatics tools. So we use bioinformatics tools to look for essential proteins within it. And of course, through this paper, we discovered about 10 essential proteins. And from there, we found that about 3 of those can be druggable as well. So there is a process that we went through. First, you have to have the database. You can have a database, then you choose a specific strain, which is going to be a reference strain within Enterococcus faecalis, within the Aedes. And from there, you are going to remove paralogous proteins, all those, because they'll be redundant, so you don't want them to be there. So only you want to remove all the paralogous proteins. So all the amino acids less than, within paralogous proteins less than 100 amino acids, we are removing pathogen-specific proteins. Then you look at essential genes within them using specific databases, like a database for essential genes. And within there, we also are going to look at virulent protein screening. So from there, we go to non-paralogous essential and virulent proteins. So through all these processes, we went all the way to identify specific essential druggable proteins within Enterococcus. These are proteins that after we have gone through all these processes, we know they're not going to target, if we, for example, because that's our next step. Our next paper is we are going to be looking at molecular docking. So we are going to come up with compounds that are going to target these specific proteins. Then from there, it means we're going to go into wet lab now, try to test them to see if these proteins are going to work. So looking at this is how important bioinformatics is. Bioinformatics is very important because we can do all this before we go into the wet lab, before we go into the lab itself, and we do experiments, and we spend so much money. We first check, are these proteins really the best we can target? So we are going to make sure that they're not going to target the gut microbials. They're not going to target that the gut microbiota is the one which is inside ourselves. We know they're very important in our intestines. They help some of them in manufacture, for example, vitamin K, which is important in blood clotting. So we don't want to kill the gut microbials. So we want to make sure that when we are doing whatever we are doing, we make sure that those proteins that we're going to target in this specific bacteria is only those proteins in there, but not any other. And going forward, we remove and target proteins from everything else. So we don't want any other proteins also to be targeted as well. So then from there, we did broad spectrum analysis. So we are saying, okay, this is the bacteria that we want, and we want this bacteria to be taken out. If we design a drug against this particular bacteria, can it also target other bacteria? Can it become broad spectrum that we can also even kill other pathogenic bacteria that affects? So we do the broad spectrum analysis, and we found that yes, if we are going to target these particular proteins, in addition to our bacteria, we can also be able to target other bacteria that are causing all sorts of problems as well. So that's very good. Then we move forward. So we do the host pathogen interactions, drugability analysis. So we are going to check that everything is fitting in. We do the drug bank analysis, therapeutic target database, and then we check the typical physiological physiochemical parameters for druggable proteins. So all this put together, it helps, and we have done all this how we are sitting on the computer. So it means it's something that we can do. We can find that sometimes getting money, getting funding to get your research funded is sometimes very difficult. So people get stuck. Sometimes to get, let's say, mass spectroscopies, they're very, very expensive. Transmission cryo TM, I remember one time I was talking to a professor at University of Oxford, and I was trying to convince him that we should be able to have a TM, transmission electron microscope, and in Malawi, for example, and you can be able to use it for a protein structure analysis. And he was telling me that your institution alone is not going to work. You need to have almost the entire region. So you need to go to Tanzania, you go to Zambia, you come to Malawi. So you form a team. Then, because even here at Oxford, we have at least 5 departments that are actually just looking after one instrument. So because of all those sometimes of hurdles, using bioinformatics tools, we can still be able to do some work and be able to come up with something. That's the important part of bioinformatics, that we can be able to do something and from there come up with something. So this is just another example. So depending on time, I think we can go to this because if you have discovered we have got a protein, so this is a application of bioinformatics. So I can try, maybe let me just see if I can share an example of some of those tools that you have seen there, how we have used it. So we can come to these two, and then we can share that so that we can all be able to appreciate some of these tools, how we use them. So for example, I'm sure maybe we are all able to see this. So this is a comprehensive antibiotic resistance database. So this particular database, for example, you have that Enterococcus faecalis, and you want to claim, or you have found a good example that you have found a new strain altogether. It has been sequenced. Now you want to know if this new particular strain, the sequence is available, say on NCBR, if this new particular strain can also develop drug resistance. What type of drugs can this particular new strain be resistant to? That is before even it becomes very bad in the hospitals. So this is a new strain. Then you want to check, can this be checked if it has got any drug resistance within it? So you can come here within the analysis. So you have got a new genome, and from the new genome, then you can upload this entire genome. This is the entire genome of that particular organism. So you have got the entire genome of that particular organism. So in this case, is our Enterococcus faecalis. So in this case, we have no idea if this particular strain is resistant to any drug. It's a new strain, just recently discovered, and it's out there. What type of antibiotics it will be resistant to? That's our aim now. That's what we are trying to achieve. That's before we can do all the drug analysis. So you take the entire genome. That genome is downloaded from Edgar's database. So you are downloading in Edgar's database. You get the entire genome. Then from there, you start doing the analysis. You are trying to analyze what type of antibiotics it will be resistant to. So it was also part of that paper. We did also this so that we can say, yes, this particular strain that we are looking at, it can indeed develop antibiotic resistance. It has good antibiotic resistance in it. So using the bioinformatics tools. So from there, so here we have, these are the results. So this can be a new strain. These are the results. So we know that it is resistance, detection, protein, gene family, glycoprotein resistance gene, cluster of VAN-T, glycopeptide resistance, antibiotic target alteration, and this is the sequence similarities. So we know that it can be resistant to specific antibiotics, and these are antibiotics, and the mechanisms it can use to resist are also being shown. So we can be able to see this is the antibiotic influx. So all this information is very important using bioinformatics tools. This is how bioinformatics tools become very, very important. So that you can have a new sequence from a new bacteria which you have no idea what is capable of. This is just a new strain of a pathogenic bacteria. Let's say tuberculosis has developed resistance, but we don't know what's going on. So we can have that. We can do exactly what I've just done now, and through that you can be able to see that this particular bacteria can be indeed resistant to this. Then from there, the next step, of course, will be saying, how do we combat this antibiotic resistance? And as I've said, in our case, we are saying, let's look for a new drug targets. That all these drugs, all these antibiotics that we have, this particular Enterococcus is resistant to. So what do we target next out of all these? So we can use bioinformatics too. That's what we were doing at that stage. So then we can go back to our presentation. That was just to show you, that's just an example of how we have used this bioinformatics to analyze. So the next one is we use also the same bioinformatics tools in detecting the biochemical pathway. So we know that this particular bacteria, Rhodomonas procaris, is the bacteria which is involved in sustainable bioremediation. We know that experiment has been done, and the data is very, very variable. In fact, this bacteria is involved with so many other things apart from bioremediation, but we don't know the biochemical pathway. So from there, we came up with this paper using bioinformatics tools to try to find which parts of this particular bacteria can be used to do bioremediation. So those are the stuff that we have been doing to come up with to use bioinformatics tools. So in conclusion, importance of bioinformatics is increasing rapidly in advances on omics, i.e. genomics, proteomics, and information storage. As more information storage are coming up with new tools that we can use to store information, transmit that information very fast, and process that information, bioinformatics also is advancing at the same time. So tools as well are becoming more and more, and starting from just basic analysis of a sequence to really big web server sequences. So some of them, in fact, all the tools that we use, for example, from here, all these tools that we have used to do this particular research were free of charge. So all of them are freely available. All these are freely available. So there is no tool there that we actually paid for. All these tools are available, and they're also coming from very well referenced papers. So we choose all these tools based on also their publication records. So say, this tool has been published in this very good journal, so this tool is very nice. We can actually use it, has got a very good reputation. Why not? Let's use this. So all these tools, there's none of these tools that we have to pay for. Some of them, of course, even Blast itself, you have to pay depending on how much you are going to be using. So if you use a lot of data, then you can go all the way to have your own standalone web servers within the institution that you may have. In fact, it's very much encouraged that we should have bioinformatics labs. So bioinformatics labs within an institution, no matter how many, maybe even just few PCs, 10 or so, is very important to use those, and then you can install some of the softwares within those for analysis of sequences. So for those that are interested to read more on this, you can always go through these publications, Bioinformatics, Genomics, and Proteomics tools for drug design. It's a very good book that you can go through and check on that. And also Bioinformatics Basics also is one of those as well. So basically, that's the idea. So overall, the aim was just to give you an appreciation of how important bioinformatics is to the analysis of genes and proteins. And this is just a quick overview of what bioinformatics is capable of. But any questions, we are very much free to ask after the other presenter. So thank you very much for your attendance. Any questions, please feel free to post it in the chat. I hand over to the chair, Dr. Yusuf Agabi.
Okay. Thank you very much for that, Dr. Rodrick, for the powerful breakdown of bioinformatics up to its applications. We appreciate you for that insightful presentation. Next, we'll hear from Dr. Wadzani Dauda, who will be taking us on phylogenetic analysis. Before then, if you have any question, please note it in the chat box at the end of the sessions. We'll take them one after the other. So now we hear from Dr. Wadzani on the phylogenetic analysis. You'll be taking us on hands-on demonstration here.
Okay, just before then, we would like Anayo from University of Jos, Nigeria, to read the citation of Dr. Wadzani Dauda. You're welcome, Anayo.
Thank you very much, Dr. Yusuf, for that opportunity to read a brief citation of Dr. Wadzani Dauda Paunan. Dr. Wadzani Dauda Paunan is a researcher, academic mentor, and a life coach currently serving as a faculty member at the Federal University, Gashua, Nigeria. His illustrious career is marked by over 120 scholarly publications, reflecting his unwavering commitment to advancing scientific discourse. In 2025, Dr. Dauda was ranked number 10 by Skival and Scopus in 2 different fields, from biological sciences and agricultural science, among 500 prolific authors from 58 different countries in Africa. Dr. Dauda holds esteemed positions, including editorial board membership for Frontiers in Genome Editing, +1, Springer Nature, Francis, and Elsevier journals, recognized for his scholarly eminence. Dr. Dauda has received accolades and grants from prestigious organizations such as the Bill and Melinda Gates Foundation, UNESCO, Originarium, American Phytopathological Society, American Society of Microbiology, Department of Biotechnology, India, World Academy of Science, twice, Italy, Tertiary Education Transfer Fund, and prestigious M.J. Duramalaka Merit Award for Young Scientists in 2022. Due to the significant nature of Dr. Dauda's research findings and his noteworthy achievements, it is unsurprising that he has been offered postdoctoral positions in esteemed academic research laboratories at Lausanne State University, USA, Hebrew University of Jerusalem, Israel, Maastricht University, Czech Republic, and Indian Institute of Technology, Mandi, India. His extensive research interests encompass diverse domains, including disease detection, mycotoxicology, agricultural microbiology, genome editing, and bioinformatics. Dr. Dauda's intellectual versatility is further heightened by his membership in renowned societies, such as the American Society of Biochemistry and Molecular Biology, International Society for Computational Biology, International Society for Molecular Plant-Microbe Interaction, Society for Applied Microbiology, American Phytopathological Society, American Society of Microbiology, the Nigerian Young Academy, and the Royal Society of Biology, UK. He holds the esteemed title of Fellow of the Society for Plant Research. Dr. Dauda is the Director of Research and Development at Academic and Grant Writing Enclave H. Dr. Dauda has been invited as a keynote speaker at various national, international institutions and conferences, including the UNESCO International Center for Biotechnology in Nsukka, Nigerian Doctoral Academy of Nigeria, University of Nigeria, Nsukka, Nigeria, Doctoral Academy of Nigeria, National Open University of Nigeria, University of Jos, Nnamdi Azikiwe University, Anamala University, and several others. Dr. Dauda's collaborative endeavor extends globally, fostering intellectual exchange with research groups in leading universities. In essence, he stands an exemplary of scholarly excellence, illuminating the path of scientific inquiry, leaving an indelible mark on the annals of academia. Dr. Dauda, a dedicated researcher who values the importance of family, is happily married to engineer Deborah Dauda, and together they are blessed with 2 sons. Audience, please permit me to welcome the person of Dr. Wadzani Dauda Panam. Doc, the floor is open for you. Thank you.
Yeah, thank you so much, sir. I don't know if I'm audible. Yeah.
We can hear you. We can hear you.
Yeah, I can hear you.
All right. Thank you so much, everyone, the team, the organizers for staging this great event, and they hope that of course, I joined where the second speaker was rounding up, and saw the quite great work they presented, and I guess the speaker before him must have done a very great job also. And so hopefully I think this might be the closing. So it's great to be here, and I hope that we gain value in our discussion today. So without much ado, I'll be speaking on this topic I just tried to frame so we could learn and share together from the few works we've done in computational biology. And so some good introduction discussion has been done already on bioinformatics by the previous speaker, so I will not want to really overflog it. But then just a quick recast, the concept behind the entire biotechnology is because of the evolution of the next generation sequencing machine and instruments that have evolved, and they have made the ability to decipher not just the morphology or the physiology of animals or plants, but that the genome of each organism could be looked into, such that from this chromatogram file you see meanings and interpretations. And of course, if you remember the Nobel Prize that was given to Alfensen, it was because of his innovation behind sequences correspond to functions. And that's the basics of bioinformatics, actually. The fact that from sequences, you could tell functions of literally every living thing. So wherever they are living things, you will find sequences. And that is the concept behind the bioinformatics as a discipline. All right. Some of the common tools that are generally used, which the previous speaker have talked about already, and I will not, like I said, I won't overflog them again. But then what I just needed to bring home is what bioinformatics is not. Bioinformatics is not just using computer to speed biology. It's not just applying computer algorithm to biology. All right. It's not just the accountant of genome data. No, it's your ability to make sense of a data. And I remember I think that should be the second postdoc fellowship I got. It was the grant. I think it was at the Hebrew University, Jerusalem. I saw, I think it was on Twitter when I was a bit active on Twitter at that time, and Professor Yavo dropped a post that there is this particular grant at the Museum of XYZ in Jerusalem, and the grant call is for a postdoctoral research fellow to come work on this project. And then I was rounding up my PhD, so I was really applying for postdocs. And so at the time I saw it, it was exactly on the 16th of April. Yeah. So at the time I saw it, I checked the comment section and I saw exactly 126 comments from people who have dropped message. Prof, I've applied, Prof, I've applied, Prof, I've applied. And I was like, oh my God. So what are my chances that if I apply, and I'm even seeing it late? And so, but somehow what I did was I went to the prof's lab, looked at his lab, what they're doing, and saw the kind of research he's doing, then went to the funding agency, checked the grant call on their website, understood what they were trying to do. And so from there, I came up with the idea of doing an evolutionary analysis of ancient wheat rust pathogens in Jerusalem. And so in fact, then my references that I was using were old, really old references. I'm talking of, for those of you that are Christians, if you read Bible, you see in the era of Moses, even Muslims, in Moses' era and the mountain Sinai, so those were the kind of references I was citing. I was citing Quran, Bible, other publications, because they were actually in the museum. They had old pile of, wheat plant from those thousands of years ago. You can imagine how these people are very intentional with preservation of history. So they have it all stocked up in their museum. That was on the website. So I came up with an idea. Okay, let me come up with an idea where we can use biotechnology, do reverse, amplification of the sequences using some selected primers across the major wheat rust. And through that, we could be able to trace the evolutionary relationship behind and the connection behind those ancient wheat rusts with the current wheat rust thousands of years later. I'm telling you, that was just it. And I got the grant. I think the grant was, I've forgotten this thing now, but of course, eventually I didn't end up go because of one or two reasons. So, but that is just how these things happen, right? So bioinformatics is the creative use of computer to define and solve central biological puzzles. That's the idea. The computer becomes an hypothesis machine making predictions to be tested at the bench. So that's why, like my former colleague that was talking, you see a lot of works that they talked about in their group. And so it uses the power of prediction, right? Features of bioinformatics to solve problems. It's less time consuming. That's a plus because sometimes you can do a whole analysis and you could get stopped at some point in time. And so, so that's just the thing. Less labor oriented and less expensive, and narrows down the search. Not complementary, rather supplementary to the traditional work. It helps to bring in precision. And lately, I've been seeing a lot of trends on application of AI in bioinformatics. In fact, so this is what basically happens with it. So what are two key branches of bioinformatics is the development and implementation of tools and database. That is one key component of bioinformatics. The second component, development of new algorithms and statistical formulas. And of course, the third component, use of tools to generate biological information and manage them. So what we do with life sciences, basically we dwell more on the third one. The other first two, particularly the second is computer scientists that do that. They develop the algorithm, mostly we use them. Because you would say you want to go back to start learning how to write algorithm to develop tools and all of that. Usually we learn how to apply them, right? So because the basics behind it, of course, like I said earlier, is the central dogma of bioinformatics starts with sequence similarity, which corresponds to structural similarity, and of course, the functional similarity. And this same basic concept, as you see, was what gave Hansen the Nobel Prize, I think in 1979, if I remember. So this is how these things happen. From match query, then you try to see the match. From there, primers have been designed and applications have been done for various sequences and various analysis. So how bioinformatics exactly works is this: learn and generalize, organize and integrate the data you're getting, make predictions, and from the predictions, make simulations. And so the idea behind it at the end of the day is to find the meaning of hidden information in the sequence. That's what it does. That's the purpose. That's what it aims to achieve at the end of the day. From where we get all those sequences, of course, from the sequence database. So these are some of the basic concepts we just need to understand before we talked about it. In our research group, one of the work we did some years ago were to look at certain, code protein of viruses across Africa. And we're trying to really evaluate whether code proteins, yes, code proteins we knew had various functions in virus, but we're trying to really look at whether these code proteins can tell us a little about the evolutionary connection between these viruses in Africa with 16 African countries, then use Indian strain as as a control or as an outlier to really compare the relationship. And we could see from this result here that literally all of the other African strains were aligning in a particular clade, except that from South Africa with East African Malawi virus, where they were clustered together. And of course, the Indian strain was there as an outlet, confirming what our earlier hypothesis. This was published in Gene Report. And we also recently we began to do I think some 4 years ago, we got a grant where we're working on certain bacillus strains in cassava wastewater in Lagos, Nigeria. And the work we did a lot of expensive work where we sequenced about 6 bacillus genomes, and I did the entire computational works in that project. That was actually my role. I was a bioinformatician in the project, and we did a lot of work. In fact, just this afternoon, the 6th paper from that research was published. This was the second, where we did the genome announcement, did the little characterization of the bacillus strains that we obtained from these locations. And this was the third genome where we did the analysis and tried to look at what are the similarities across it and how does it connect to these wastewater locations. And so recently also, which was just released today, where we did a comparative analysis, and we saw that indeed, actually, these bacillus strains had very strong connections. Currently, what we are doing now is we're looking at antimicrobial resistant genes in it. That's the final, I think that should be the 7th paper we're rounding up in that project. It was sponsored by Bill and Melinda Gates Foundation. And so what is biological database and how do you get it? Organize the body of persistent data where you query, from your query, you trace the components and you make sense of your data. And so this is how these things happen from the sequence B. Then you look at it, the structural proteins, which I think the other colleague talked about it earlier. Nucleotide sequence, then you have them translated to protein sequences. Depends on what exactly is your biological question. Then moving forward also, this was also another work where we're trying to look at various strains across various, this was a particular fungi, and we saw the connections between how these strains are interconnected and where they are located exactly in the chromosome. So one of the principal tools that is being used in bioinformatics generally, the fundamental that everyone knows is, of course, the NCBI. That is the goal to get. And recently I was trying to do some work and I saw a lot of updates that have happened in MCBI. So one of the first places you would want to go to get your hands dirty in bioinformatics is actually at the NCBI, right? And so to our discussion today, what exactly is phylogenetic analysis? What do you do? And if Mega is the one, there are, I think, about 30-something different tools that you can use for phylogenetic analysis. You see this tree. Now, when you look closely at this tree on the slide, what you observe is you see that from a single branch, it has, from a trunk of the tree, you see several branches across. Branches have sub-branches, then sub-branches, then sub-branches. So that is the idea behind using phylogenetic analysis to really see ancestry, synteny, comparison, alignment. These are the features you look at when you trying to check the phylogenetic relationship across organisms, right? So I will talk about Mega because that's a common one, and I hope today I will have there will be a short practical session that done by Dr. Elkanah. He's the team leader in my research group, and he'll be taking us a short demonstration, just show us how this is done, right? Yeah. So Mega is actually one of the common tools that are being used in the molecular evolution. You know, it's been there since 90s. You know, the developers where they did extensive work, and interestingly, it's free, right? Whether it's transcriptomics data, proteomics data, genomics data, all kinds of data are being loaded in NCBI, and it's a good go-to. That's the first place to actually begin from, right? So these are the steps in doing it. Sequence data collections, where you start with collecting your sequences, depends on where you are, or sometimes there are sequences that you sequence yourself using whole genome sequencing, like the one we did in the project I talked about earlier. Sometimes you would get it from the NCBI itself. So it depends really on what exactly is your biological question, right? So because at the end of the day, you realize that the concept of science and what it happens, it has a lot to do with your intelligence. Yes. I had a class some last month or thereabout, where I was taking them on a series on grant writing, and I was talking about AI and prompt engineering, and I said the era of everything, literally everything begins with your intuitive mindset and how intelligent you are to decipher certain things. These things no AI can do for anybody. So the ideation and how you think, if you reason one-way traffic or one-way round, you can't see innovative or ideas behind it. All right. So these are the basic steps actually. Where from there you do sequence alignment, and from there you select your model. Depends on what exactly you're looking for, right? But these are the general features of every phylogenetic tree that you would ever do, right? Construct your tree, and of course, tree visualization. Bootstrapping depends, but generally, bootstrapping is very critical because it really tells how statistically accurate your analysis is. And that's why, in fact, some of the journals I review for, I edit for, once I see a sequence and you didn't put bootstrapping, the bootstrap values, generally, I just know that you just gathered them and just brought them. And it can just be anything. You could as well just match them. But the bootstrap value really tells how statistically aligned, perfect your works are. So these are some of the starting point, like I said earlier. Begin by DNA or protein sequence retrieval, and it proceeds from there. You obtain and validate your sequences, and you would want, because sometimes one of the questions I do tell people is, when you sequence your sequence, the first thing is you try to submit it to NCBI. They have well-robust system to tell you whether I've submitted quite a number of sequences, protein sequences, DNA sequences, whole genome sequencing, transcriptome, all kinds of sequences have submitted to NCBI. So one thing I've realized is that you just can't fabricate, manipulate the system. No, it's just there. You send it, they will do a query, search it, and tell you whether what you've done is right or wrong. And it goes through that scrutiny, every sequence that has been submitted. And so that's why I remembered one of the work we did one time. We were thinking it was a particular fungi, checked upon when we sequenced the part, that was using a partial sequence. We realized that it was a completely different organism altogether. And so these things happen. Multiple sequence alignment, which is which has in the NCBI, the alignment explorer, usually cluster W, is the one most of the algorithm that is used, or sometimes Mzuzu. So moving forward, because if you look at all of them in its entirety, the phylogeny, the construction method generally follows this either neighbor joining method, maximum parsimony, where, okay, if you are dealing with maximum parsimony, maybe you are trying to look at the fewest evolutionary changes as precise. When you are trying to be a little bit more precise, especially when you are dealing with sequences that are really close to each other, generally we use maximum parsimony or maximum likelihood, of which is at this point you are looking at, when you are looking at various sequences that, okay, maybe this one is from South Africa, the other one is from Italy. So generally, when you, from the analysis you've done, and they are very fast regions, you use maximum likelihood. Quite a number of course, these algorithms, the pipelines depends on your biological question. So generally, how you know which one to use, it depends on what was your biological question in the first place. So before constructing your final phylogenetic tree, you have to look at certain criteria, right? Model. And I need to say this really, really quick. It is sometimes you see a lot of people, when they do work, they just match sequences, just did any sequence alignment. They don't even understand which statistics to use because the person is not willing to read. You just pick sequences and just run a phylogeny on them. I hope if we have the time to do the Dr. Elkanah does a little demonstration for us today. We have the time. But those are the things you really want to check. You see, the core of every experiment is not really just entering and just doing and doing and doing and doing. I think I was talking to someone today and I was telling the person about different kinds of soldiers, right? Or military persons. There are soldiers who are snipers. Snipers, their bullets are calculated. Yes, their gold standard is precision. How? Because if a sniper is hiding behind a rock or somewhere, and you remember when that guy that was attempting to kill President Trump, he missed the target and he died. That's how it happens. Once a sniper misses it, you have consequence. So a researcher is supposed to be a sniper because sniper will have to calculate angle. I have a lot of friends. One of my friends just became a general, I think last year, my childhood friend. So the training of a sniper is different from the training of a general infantry army, because an infantry soldier could just, knows how to navigate and all of that and all of that. A sniper, the gold standard is precision. So before you really start any bioinformatics analysis or whatever, you really want to understand what exactly are you looking for? What's the biological question? What's the hypothesis? What's your thought? What's the research question? How do you know if the hypothesis is going to be positive or negative? And how do you handle that? All right. So the various tools that have, in fact, quite a number of tools that have been developed recently on tree visualization. This is one of our recent work that we did, where we looked at various Trichoderma species. Trichoderma is a fungi, and through phylogenetics analysis, we realized the common alignment across some of the specific Trichoderma species. The paper was published this year in Frontiers. So, and we saw the similarity across some of the few selected Trichoderma species, and from there we did, SEM analysis, and from the SEM analysis we narrowed down on two and did an antimicrobial analysis of assay of them, and very interesting work. So the thing here is so that's what bioinformatics helps you do. It helps you to narrow down on your search. It doesn't replace the main wet lab work, but it helps you to narrow down on your search and be able to make sense and be able to tell, okay, what are the things to now specifically focus on, right? The advanced features of bioinformatics, evolutionary relationship, time tree wizard, ancestral reconstruction, selection test, and gene duplication analysis. This is especially done when you are looking at mutation across sequences, and of course, whether they are sequences of plants or whatever it is. This is one of our projects that we did some years ago, where we published quite a number of works in that. There we were looking at really evolutionary relations of organisms, and and we saw how this cytochrome P450 diverge and causes the distinctions across these organisms, these fungal organisms, that although they are from different species and genera, but one thing still remains: the distribution and the multiple abundance of cytochrome P450 in this. So this is the case study that I hope Dr. Elkanah takes you through briefly in the next 10 to 15 minutes after this talk. But where he will take you through it so you could do well to start downloading Mega, and to just run through briefly the next 10 to 15 minutes, if there is time anyway, because I learned the question and answer sessions also. So the way you try to track viral transmission, where you imagine a scenario where you know a virus, and as a researcher, you want to understand the origin and how you use Mega to do that. So I hope we have the time for that anyway. So he will walk you through the Mzuzu, the alignment, sequence retrieval. He walk you through how to retrieve the sequence, how to do the alignment, how to select the module, and how to do maximum likelihood, how to, map in your bootstrap values, and how to really interpret it. So, like I said, if we have the time, we could do that today, but if not, maybe another time. So, so this is how you want to launch Mega. You open the file, which I said you could download it in fast file. You select the sequences and run multiple analysis using cluster W or Mzuzu. Depend, like I said earlier, you trim sequences and run multiple sequence alignment. You now go to do your phylogeny. And of course, the polishing of your tree, that is now depending on how you want it presented. And the quick way to know how you want it presented is by looking at other people's work and how they presented. So this is a typical phylogeny. The sequence, you see how they are grouped into 3 clades. Strains here, you see how those in Asia, the Arctic related and the cosmopolitan, how each of them are further subdivided into 9 subclades. See, so this is how you know which one is in a clade, which ones are subclades, right? So you try to perform a phylogenetic analysis of the query sequence and draw phylogenetic tree and polish it. This is, I hope, the task we hope to do. And of course, this is a summary of the entire story. So thank you so much, ladies and gentlemen. I think time is up. Yeah, thank you.
Okay. Thank you so much, Dr. Wadzani, for that insightful presentation. Yeah, Ben, over to you.
Okay, thank you very much for that. Now we'll move. We'll move over to the brief hands-on session just for 10 minutes for we to see briefly
no,
what
Ben,
goes on there.
No, I think we can go. Let's take the questions first.
Okay.
And then we can round up. And then after that, we'll take the answer. Those who want the answer can stay on, while others who want to leave can leave after the question and answers with Dr. Agwale and the other speakers. Thank you.
Okay, thank you very much for that correction. Okay, our very first question here will be taken from the questions recorded from our Google form. So the first question here is how bioinformatics can be utilized to alter code that will result in the change of human behavior. So that's the first question. And another question is how in silico can be applied into real world situations. Then we have its benefits to, let's say, someone in academia full time into teaching. So how does it benefit such a person?
Okay, so, what about the questions on vaccine and all that from the first?
I'm trying to access the chat, but I'm unable to do so. Maybe you could take that.
Okay. Okay, maybe let's take these ones. I think Dr. Katete, can you answer these ones? Then the person that posted questions on relating to vaccine in Africa or related questions can put them up again for Dr. Agwale to answer.
Yes, I can come again on the first one, the first and the last one. I think the second one I was able to get it about the in silico application in the real world. But the first question, it wasn't very clear.
Okay. The first question is how can bioinformatics be utilized in order to alter code that results in the change of human behavior?
Okay, if I get it correct, is how we can use bioinformatics in the change of human behavior. We can use bioinformatics to change the human behavior.
Yes, sir. Yes, sir. That's the same.
Okay, so how we can use bioinformatics to change the human behavior? That's the question. I think the way we use bioinformatics so far, for now, is that we use as a launch pad. So we are using a bioinformatics as a launch pad. Before we can do so much, as I say, that you can go into the lab, you may find that you have spent so much money doing specific kind of experiment, but just realize, as Dr. Agwale said, that they go for preclinical and clinical trials, for example. You may find that to go all the way to clinical trials is very, very expensive from initial research. So to go all the way to clinical trials is very expensive. So the initial part is to do most of the work on the computer, and from there we can narrow down our searches that, okay, this is the correct, what we are targeting, this particular protein, for example, we are targeting is the correct one before it's too late, because we may find that we have spent maybe 5 years down the line, even sometimes we can go the 10 years down the line, and you find that you go to the clinical trials, you find that your drug is actually having a lot of side effects, simply because it's targeting all sorts of other human proteins. So the way we can use bioinformatics in terms of changing human behavior is to help people in general, to sensitize people that, let's say there's a new variant of COVID just now, it has just come out. Using bioinformatics tools, we can be able to know how virulent it can be. Virulent factors are there. So we can also do bioinformatics and we can know that this particular strain of COVID is very virulent. Then we can actually tell the people that you need to start putting on the masks now. You don't have to wait for it to become worse. As far as we are concerned, this particular strain is not good. So you need to change your behavior. You need to change the way you are treating, you are doing things before it's too late. So we can use that. That is even before the particular strain has spread across the entire country. By just looking, by the time it spreads and do all sorts of damage, we have at least some basics idea of what it's capable of. And we ourselves, we are very good at doing that. We can be able to check that and tell that definitely this particular, for example, if we look at, as other presenters have talked about the GP1 the HIV. The HIV is very good at glycosylation. It glycosylates itself a lot. So if the virus is mutating proteins, is bringing in amino acids that are highly glycosylated, that are going to be glycosylated, it means we can know that this particular this virus at this stage is trying to hide itself from the immune system, because glycosylation helps these viruses to hide from the immune system. So we can be able to know that okay, depending on what it's doing, it can do that. So the other one was about the in silico application into the real world. So it's the same based on that paper which I presented, that the initial stage was to isolate specific essential proteins. Essential proteins are those proteins that this bacteria cannot live without. It has to have them. If those proteins are not there, then it's going to die. There's no plan B there. So it can't live without those proteins. So identify those essential proteins using bioinformatics tools. Then the next is to say, what type of compounds, that is, call it molecular modeling and molecular modeling and docking. What type of compounds are out there that can actually target these essential proteins, and then we can create a cure. So that is the second step. The third step then into the real world is now say, okay, I have discovered this new compound. This could be the way people use any herbs. Let's say moringa. Moringa could have specific active ingredients that can target certain things, and maybe bacteria can have some antimicrobial properties. So we can say, okay, this particular antimicrobial property, it can target this particular essential protein without any side effects. Then can we check that now in the real world? That is now in the hospital from the actual isolates. Then we check that. They say, oh, yes, it's working. Then because we have got all this massive data before us, then we can say, maybe if we do a clinical trial, hopefully we may not have side effects, and hopefully we may actually be able to clear this particular bacteria. So that is how we are moving. So we are using bioinformatics as a launch pad. It's a way to, as a starting point towards the real world now. The end result, of course, is definitely going to be in the real world. So the last question had to do with academia, but I didn't understand it clearly. Can you please repeat the question, the last one about academia?
Okay. The question was on how does bioinformatics, the idea of bioinformatics, benefit a teacher, someone who is in, yes, just a teacher. That's how the question reads.
How a teacher can use bioinformatics?
Yes, sir. Yes, sir.
Also, how a teacher can use bioinformatics, like in terms of teaching. So, in most courses, especially medical-related programs, even non-medical, but let's say biological, molecular biology programs, it's very important to include within the curriculum itself bioinformatics. Ourselves, I think in biomedical sciences, we have got the medical bioinformatics as a course is there, which we teach. So it's medical bioinformatics. I've also biomedical. We have also medical biotechnology. So within medical biotechnology, we have also some components of bioinformatics as well. But within each course, as well as you complete, let's say you are teaching proteins, it will be very good. Let's say you are teaching proteins and you are at the end, there's also some kind of mutations that you want to demonstrate. It will be very good to take the students to the bioinformatics lab and then show them that these are the mutations. Like I said, on the Swiss PDB viewer, for example, you can be able to remove, let's say glycine is a very small protein, amino acid, is a very small amino acid. Then we actually say that if we remove this amino acid, then we are putting a very big amino acid like tryptophan. What is happening to the protein structure? So those are some of the things that as you are teaching, so that it just don't just become an abstract, such that they are not able to understand what is going on. They can be able to appreciate that in the bioinformatics lab by actually playing around and mutating, creating different type of mutations, and looking at those different type of mutations, then you can start appreciating that, oh, this is what is happening. Or like one of the papers that we are trying to come up with, we look at these mutations and say why these specific mutations are making this particular variant of, let's say, COVID more virulent than the previous variant. So we can actually do that. We can say, okay, probably because of these mutations, it means these mutations are telling us that if this takes place, this is what is happening in terms of the virulence of this specific strain of COVID or HIV or bacteria. So all that can also come up just from the teaching itself, from the classroom to the lab, and then you show them that these are the things that these bacteria are doing, and through these activities, they are able to create these programs.
Okay, thank you very much for that, sir. Now I will direct this next question to Dr. Simon Agwale. Okay, the initial question that was asked in the chat, vaccine development. So the participant was asking if the current research work you are involved in, if the raw materials like about the lab in Africa, the lab that developed these vaccines situated in Africa, if the raw materials and everything will be concentrated in Africa, or a kind of partnership will be required in order to sustain it. And yes, that's the question, Dr. Agwale.
Well, as you know, the entire ecosystem for vaccine development and manufacturing has to be built. As it is currently, it's going to be a gradual process, and none of this is done in isolation. So whichever way, it has to be in partnership. There are so many components of making a vaccine. You have hundreds of inputs that you need, to develop and to manufacture a vaccine. So it's not just manufacturing the vaccines. The other inputs also needs to be manufactured. Initially, you have to depend on importing those inputs because there are no manufacturers on the continent. And that is what we are pushing as African Vaccine Manufacturing Initiative, that people shouldn't just focus on making the product, but also focus on making the inputs that are needed to make a product. I'm happy and excited to know that we have bioinformatics experts on the continent. This is very, very encouraging, and webinars like this are very important to really know the capacity that does exist. I, gave a lecture recently to university colleagues in Nigeria that are setting up a postgraduate program on vaccine and drug development. And then we talked about bioinformatics And I've been working hard to use the network that we have to get experts, because you cannot set up these facilities, without bioinformatics. This is where drug development, vaccine development is going. So I'm happy that we have these experts in the country. Without meetings like this, you know what exists in the country, and then you spend a lot of energy to try to see if you can identify people outside that will come and do something that is already available locally. So I look forward to engaging with the two experts that gave a presentation on bioinformatics, and then see how we can, work together as experts on the continent, everybody doing their own piece, and then solving our own problem, because nobody's going to solve our problem for us except us. So I will like to thank Dr. Agabi for putting this meeting together, and then we look forward to further engagement. Over.
Okay. Okay. Before you go, Dr. Agwale, there's another question here from Dennis Otieno, asking you speak on the COVID vaccines, and particularly the one that was reported to cause an effect on humans, if there's any truth to it.
Well, it's difficult to say anything about the side effects. Like I said, if you look at the different vaccines, that were used during the peak of COVID, the traditional vaccines, inactivated vaccines, VLP, protein-based vaccines, the traditional vaccines that have been used over the years. But the mRNA vaccine, the viral vector vaccines, have been in development over the years, but they were used mainly during COVID, and the viral vector, of course, the AstraZeneca vaccine, as you know, eventually withdrawn because of the perceived or the side effects that were associated with the viral vector technology. So it's more of technology than because the mRNA viral vectors were never used. All the childhood vaccines are using the traditional vaccine platforms. This is the first time that we are introducing new platforms like mRNA vaccines and viral vector vaccines. So we will need time, because clinical trials are done to establish safety and efficacy of vaccines. So that's why you have from phase 1 to phase 3. Then you have phase 4, which is post-vigilance, because 3 years to do efficacy study, in most cases, is not enough to know everything about the vaccine, but it will give you indication on the safety and efficacy of the product. So once you know that, that's why the post-vigilance is important, to look at the safety over time. And then if eventually, is shown over time that it's not safe, the product is withdrawn. So it has nothing to do with vaccines, but it has something to do with the platform technology, because this is the first time we're using these two platform technologies. So we'll see what happens over time. Already, as you know, the US government is pulling back, from some of these technologies and advocating for traditional technology. So we have to watch to see what happens over time. It's not definitive. We can't conclude on anything right now, but let's see what happens over time. Over.
Okay, sir. There are more questions for you, sir. One is high infection rate a prerequisite for vaccine trial in a given population? And the other question is what motivated you to venture into vaccines, proteomics, at the time it was not most focused, at the time it was not a most focused field or venture in Africa. And the third question here is, I think you've answered the third question. So the first question is high infection rate a prerequisite for vaccine trial in a given population? And what is your motivation to venture into a vaccine proteomics?
Yes.
All of that.
If you have high infection, there's the potential, and that was why we were able to establish the efficacy of COVID vaccine within a short time, because the infection was, very high. If you look at diseases like HIV, for example, you need time, because there are existing drugs that are helping patients, and if you want to establish the efficacy of a product, you have to do a placebo control trials. One arm tests the product, the other one, you monitor them over time, and then you establish the efficacy of your product based on the number of people that are infected from both arms. And here you are stuck with the fact that somebody is in the placebo arm and there are antiretrovirals. So you have to educate the person to take their antiretrovirals. They have to use condom. They have to do that, so many. So when will you have the numbers? So it will take more time. The second is, if you look at Ebola, and Lassa fever vaccine, they are outbreak vaccines. They are unpredictable. So you do your phase 1, you do your phase 2, which is safety, immunogenicity. How about efficacy? So you can't predict when you have outcome. So the clinical trial design will be different. So we do what is called ring vaccination. So people, you have an index case, and you have people that are exposed, to the infection, and then you randomize them, and some take the vaccine, and then you have your placebo arm, and then somehow you're able to establish the efficacy. And that was the trial design that was used during the Ebola outbreak, for us to have an Ebola vaccine. So definitely you need high prevalence, of the disease to be able to quickly establish the efficacy of your vaccines. Now, we venture into vaccine because my postdoc basically was on that here in the US, and then I saw that there was virtually nothing happening in Africa, and then most of the factories at that time, vaccine factories that we had then, were focused on fill and finish. Fill and finish means you get the active ingredient from another company. All you are doing is packaging. You are filling and then selling. Now, if you look at the entire pharma industry on the continent of Africa, they are weak. You hardly see a pharma company in Africa declaring $10 million, profit, $20 million profit. Why? Because they're not involved in R&D. They don't have their products. They're buying active ingredients and then doing fill and finish. And to set up fill and finish, you need huge capex, that is capital expenditure. And then you have operational costs, and so on. So you buy this active ingredient and then add that to the cost of your equipment, which you are still paying the bank and so on. Your profit margin is minimal. It will take you years to break even. But if you invest in R&D, basically what we present, which is called the end-to-end manufacturing, you are doing your R&D, you have your own intellectual property, you are producing your active ingredient, and then you are doing fill and finish. Moderna was a very small company here in the US. It's not even their technology, they licensed it here from NIH. And they were focused on, developing it over time, and then somehow they were prepared when COVID came. And then they got lucky, the technology, mRNA technology they were working on worked. And, during COVID, a small company like Moderna with less than maybe 20, 30 staff, their valuation at that time was $200 billion. Just one product. So that's the power of intellectual property. That's the power of research and development. When you own your products, you are producing everything and so on. You have companies in Nigeria and elsewhere, they are producing 200, some of them are producing 200 products, from Panadol to aspirin to, all kinds of product names. At the end of the day, when they declare their profit, it's less than $1 or $2 million. So building factory is R&D. You develop your process, you build your factory based on the process that you have developed. You have your patent on the product, and then you are manufacturing it. You are protected. And that's where the real deal is. And then secondly, the diseases that plague us are not the diseases that plague the West. Imagine if these diseases are plaguing us, a problem here in the West, we'll have found a solution a long time ago. So secondly, it's not financially viable. Lassa, how many people are infected? It's only West African, this thing and so on. So it's not attractive for big pharma company to invest heavily to go and develop a Lassa vaccine or Lassa this. How are they going to recoup their investment? So this is what the local African companies have to deal with, have to figure out to solve our problem. So these are the two reasons that push us to working on domesticating local not just local manufacturing, local development and manufacturing. Over.
So thank you very much for that, sir. I don't know if you still have more time. There's a question here in the chat that wants you to answer. Okay, the question reads about the sequences gotten from NCBI. Does it mean that all the virus sequences for vaccine design are already in the database, meaning that there's no need for isolating new viruses? That's the question. And okay, yes, that's just it, sir.
This is a good question. It depends on the pathogen. For example, in our own case, when we were making the HIV vaccine, there was no vaccine, even experimental vaccine, that induced broadly neutralizing antibody. So the call that time was to make vaccines specific against the strain circulating in a locality. So even if you have your sequence in the database, which is publicly available because you have published, you have submitted the data. It's easy to go and take that sequence and make a vaccine, like the Wuhan strain of the spike protein. But for HIV, it's unique. So it's your sequence. So we decided that, no, this is our intellectual property. So you have to own it. But if you go to say Wuhan strain, the sequence was available immediately. It was sequenced, and everybody started picking it. So it's not is publicly available. Intellectual property will come from the platform that you use, the technology that you use to make it, but not from the sequence. You can't claim the sequence, but you can claim, and if the sequence is patented, then you have to pay royalty for the sequence. But if it is not, it is like NIH is funded by taxpayers' money, for example. So NIH, US National Institute of Health, cannot give you exclusive license for any of the technologies that they have available. So you have to get a non-exclusive license because is designed in such a way that they cannot compete, because you as a company in the US is paying tax, to the government, and the taxpayers are funding research. So NIH cannot commercialize products because you'll be competing with people that are also paying tax, to you. So in essence, this is where the real money is. When you have a disease that is peculiar to your region. I mentioned HPV vaccine, for example, that the current vaccine does not protect HPV 35, which is only prevalent in Africa. So if you update the existing vaccine, because if you want to make HPV 9, the intellectual property is already owned by another company. So it's either you license that technology from that company and pay them royalty, or when the patent expires, you can make it. If you can make it, you make it. But if you add the African unique sequence and update that vaccine, you claim intellectual property. That's where the real deal is. So I don't know if I'm clear or if you need further clarification. Tech transfer is a very complex, issue that, and this is the reason why our universities are not able to fund themselves, because, I was invited to my alma mater many years ago to give alumni lecture, and then the focus was emphasizing on setting up intellectual property and tech transfer office within the university, so that all the inventions within the university go through that, they are patented, and companies can go license those technologies, they pay royalties to the university, and that's how to sustain our education on the continent. Because if you look at the budget, the education budget, for example, in Nigeria, where I come from, it's just about $1 billion. How many universities do we have? And how many secondary schools, primary schools to share 1 billion? When you have NIH here, one research institute, their annual budget is $50 billion, which is more than the budget of a whole country. So how can you compete with a country like this? So we have a long way to go. And what universities get, look at how much they're paying professors in Nigeria. It's painful. It's pitiful. What can you expect from that kind of educational system? So these are realities that we have to face. And that is why I'm excited about these two academia, professors that gave presentation on this, and I just pray that they somehow able to get the resources that they need, to advance what they are doing, because it's a system that unfortunately there's no resources to support initiative like this. We have to continue to depend, and each donor money that comes, they have their own focus, which is not always the interest of the researcher or the country, but you are forced to do, because you are looking for funds, so you are forced to do things that are not even relevant, for people. So this is a discussion that we're having at very high level, to see what can be done on the continent of Africa, because we can't continue like this. Over.
Thank you very much, Doctor, for that, for sharing from your wealth of experience. It's really a great one. Thank you very much for providing answers to questions we asked. Okay, so there's one question, although I'll be directing it to Dr. Wadzani on bioinformatics. It has to do with the knockdown of disease long non-coding RNA. So, like, what happens really, because hearing knockdown of disease and all of that. So, what really happens in that scenario?
I didn't really understand your question, sir. Can you take it again?
Okay, if once you say the knockdown, carrying out research on the knockdown of Zig's long non-coding RNA, what does it mean?
Well, there are two things here. There are two approaches when you identify a particular gene and you want to, knock it down or repress its effect. It's either you do a gene knockdown, right? So either you do a gene knockdown or gene knockout, right? These are the two basic approaches in functional analysis now. So what happens in gene knockdown, like you are trying to repress the functions of the gene, and that's when you use the RNAi, right? Where you make an antisense region of your target gene of interest and it represses it. And generally, because for most systems, because sometimes some of those genes are either having some epistatic functions, or they're having other good functional features in other parts of the living system. So you do gene repression to repress or to suppress the expression of it, and that's when you use RNA. When you are using tools like CRISPR-Cas, you are completely removing the gene. That's what called gene knockout, right? So that is why before you want to go into, I was involved in a project, I think some 2 years ago, where we're trying to design the primer to target a particular CRISPR region. It takes a lot of carefulness because you have to understand the functionality of the gene you are targeting, its role, and quite a number of features. So it just depends on the actual function of the gene that determine what you do. And of course, if a gene is having other positive functions in other part, you can't remove it because you now affect the system. So I think that's just what I would say briefly on that. Thank you. And let me comment. Thank you so much, sir. Dr. Simon, it's great to meet you on this platform. And thank you so much. I joined, I think, when you were rounding up because I was rushing up from the lab, and also it's great to meet you here. Thank you. Over.
Okay, thank you very much, Dr. Wadzani. So I think there are no more questions in the chat. So if you could kindly take on the hands-on session.
Okay. Dr. Ben, thank you very much. Because of the peculiarity of duties on Dr. Simon Agwale, we would like to allow him to go, but want to sincerely say thank you so much, sir, for taking our time out of your very many schedules to be here to inspire us, especially coming from very limited African settings where it has been like a long walk to freedom. We hope that with your support and continued inspiration, we would do better in Africa and then also impact the rest of the world. Thank you so much, sir. We are sincerely grateful. And to Dr. Wadzani too, is a very busy person, multitasking with a lot of duties. He's not just a scientist, but he's a pastor. Like you saw in his profile, he also has a doctor of divinity, and he's changing lives and doing science with integrity. Is the director of academic.
I would like to talk to him about theology.
Seriously.
Why they are allowing the nonsense going on in Nigeria?
So yeah, he's doing a lot, and I would recommend Academic Grant Writing Enclave, one of his academies where he's doing a lot of impacts globally, impacting institutions all around the world. Dr. Rodrick has been a great blessing. Personally, he was my mentor during my doctoral degree at the University of Cape Town. He was the postdoc under the Carnegie Fellowship, where I was a Carnegie scholar as a doctoral student. And he has remained my mentor. Thank you very much. Thank you, Ben. Thank you, Anayo, and all the Phyla Biotech team, Dr. Kufre in Germany, and all our professors and participants who joined. We are most grateful. So we'll allow our speakers to go, and then we'll like that Dr. Elkanah will still take us through the demonstration. For those who want to have maybe the certificate and the records, because if you don't stay till the end, don't ask me for any record. Thank you. God bless you. And we want to say our gratitude goes to the all-wise God, the eternal, the immortal, the invincible, who gives us wisdom and even dreams on how to develop vaccines and then understand structure and function of biological entities. Thank you. God bless you, sir.
All right, thank you so much, sir. So I think Dr. Kana is, like I introduced him earlier, is the research team leader in one of the recent genomics works we did in my research group. So he's here and he will take us through a great session. So please stay tuned and then. So Dr. Kana, please, the floor is yours. I think he has been around for some time.
Yes, sir. Yeah. Thank you.
Okay, sorry, Dr. Elkanah, before you come up, we'll give just 1 minute to Professor Ajima, a pharmacist, to just give some remarks. Sorry about that.
Okay, thank you very much, Dr. Agabi.
Thank you.
Yeah, so good evening, distinguished senior colleagues and colleagues. Just wanted to appreciate all the resource persons for the very insightful talks we've had this evening. From Dr. Agwale's plenary on vaccine manufacturing was really interesting. In my department, we actually run a program in bioinformatics and genomics, but our focus has been on pharmaceutical bioinformatics. So this brought a new dimension to it, application of bioinformatics and genomics to vaccine manufacturing and development. So very insightful, and we really appreciated it. And also the talk from Dr. Dauda, from Dr. Rodrick, all the talks were very interesting, and we just want to appreciate the resource persons, and then also, of course, the Phyla Biotech team for this wonderful, putting this wonderful session together. We look forward to more of this, like Luvatui to ask for more. So please keep us in mind as you prepare more sessions in future. I'm sure all the participants have had a wonderful time, and we just want to say thank you. Thank you very much.
Thank you, Prof. Thank you. I'm sincerely grateful. All right. Over to you, Dr. Kana, and maybe Dr. Dauda.
Okay. Good evening once more. Thank you, everyone, for the opportunity, and especially the organizers, too. I want to sincerely appreciate my mentor, Dr. Wadzani, for the privilege. He has been a great mentor, a life coach, and a teacher to so many of us. I'm one of the very few that were privileged to be under his tutelage in genomics and bioinformatics research, or just one of the so many things he's into. And to start with, this is just a practical demonstration of what he has covered already, talking about a phylogenetic analysis using Mega as a tool for that. So if you launch a Mega platform, you are going to see this. Sorry, I don't know if my screen is visible.
Yes, it is. I can see it.
Okay. Thank you. Sorry. Where is my launch pad again? I'm sorry. Opened the Mega before, and I mistakenly closed it. So once you launch the Mega software, of course, one of the recent app is an offline package, which is Mega 12. You have Mega X and so many other versions of the Mega, which is molecular and evolutionary genomics tool for analysis, and it can do a lot of things, as it has been said by Dr. Wadzani. And the tool is actually freely available online. You can do a Google search and you can download it very freely to your system, which you can use it offline. Once you have it, except if you have your sequences, you don't need to go online for anything. You'll be able to run all your phylogenetic analysis and polish it as expected from the comfort of your room. Sorry, I'm having a little challenge. I don't know whether the platform is still coming up. I mistakenly closed it. So I will be using Mega 12. That is what I have for my system for now. So, and there's not much difference between even the Mega X, Mega 12, and other versions of the Mega when it comes to the interface. What is happening now? So once you have your sequences ready, as it has been demonstrated, whether from NCBI or the ones you are able to download by yourself, all that you needed to do is to prepare them in a faster format, or to download them in a faster format for you to be able to run the analysis. And one beautiful thing about Mega is that you can either use the protein sequence or the DNA sequence to be able to run your phylogenetic analysis so as to study the evolutionary relationship of these things. What is happening? Sorry, let me stop sharing and see if the Mega software can be up again. So thank you. I think it's up now. Let me share again. As I've said, please, if you're able to launch it, you're going to see this. I don't know whether the mega software is up now. Molecular and evolutionary genetic analysis. Can we see the platform?
Yes, I can see it.
Yes, thank you very much. So this is the first thing you see when you want to do any menu or an icon for alignment, data, models, distance, diversity, phylogeny, user, and a lot of other things which you can actually run on this particular software. So just like I've said, once you have your sequence ready, the only thing you needed to do is to come here, because the first step, as we have seen in the presentation of Dr. Wadzani, you've seen that you're going to do what we call multiple sequence alignment. Because if you want to run phylogenetic analysis, you want to see the different relationship or the evolutionary history be among a lot of sequences. So you're going to do alignment, and if you're going to do alignment, you're going to click on this alignment. Once you click on alignment, you're going to have different options. You can see one. The first option is whether you want to edit or build an alignment, or you want to open a save alignment session if you have it already on your system save, or you want to get a sequence from, you do a blast search, which this one can automatically connect you even to the NCBI to get your sequences. So for my own, I'm going to say I want to edit or build alignment because I already have some sequences for this very tutorial because of time. So you click on that, edit or build alignment. Once you do that, this is a prompt that is asking me to specify what I want to do. And in this case, I want to create a new alignment because we're starting from a scratch. I don't want to use a saved one. So this is create new alignment, is already highlighted. I put OK. Then it's going to open a platform for me and it's asking, what am I using for my alignment? Is it DNA sequence or a protein sequence? And of course, the one I've downloaded roughly is a protein sequence. And now this is where it is allowing me to be able to import or to paste my sequences as it can be. So if you are still seeing my screen, you can see that here I have a sequence, and this is a sequence I just downloaded yesterday evening, or roughly on some insulin. Of course, we know that it's human insulin, Homo sapiens insulin. So I decided to just take some few species of different organisms. As you can see from here, there are ones from Homo sapiens. There are other insulin from rats. There are other ones from gorilla, and some other insulin from cow, and even pigs. I just put at random to download some sequences, and I think there are about 50-something sequences in FASTA format that has to do with human insulin, which I want us to run the phylogenetic analysis. So all I needed to do now is to select all these sequences. You can highlight them all. If you can still see my screen, you can see everything has been highlighted in blue. I copy these ones. And once I copy this sequence, I can just come to my platform on the Mega software for me to be able to paste them and allow me to continue with the analysis. So once I come here, you can see here, we are already seeing sequence 1. So if I just put Control V, I can paste all those sequences I copied from that notepad. And if you can see, that everything has appeared here as the sequences that we have downloaded from the NCBI. And once you have this, remember, he talked about different platforms for multiple sequence alignment. Of course, check here, you're going to see Cluster Omega, which is one of the algorithms for multiple sequence alignment. And you also have Mzuzu algorithm for multiple sequence alignment. But first, I have to remove the first sequence that was there by default before pasting. So if you scroll this to the last one, you're going to see number 60. You can see this one, there is no sequence here because I only pasted on the existing one. So I'm going to right click on this and delete this last one that I selected. So on an alignment, look at it, align this using a protein alignment because it has already detected as a protein sequence I provided. So you click on a, this is giving me for my Mzuzu alignment. I'm using Mzuzu algorithm for the multiple sequence alignment. So for beginners at this point, we don't even need to change anything. You can use this default setting and click on OK for it to proceed with the multiple sequence alignment. And this is what it is going to run through. And as this one is running, because of time, I've already saved an alignment version of this particular treatment so that you're going to see how the aligned result of this multiple sequence, about 59 of them that I've actually imputed, will look like. So because of time, permit me to stop this and cancel this multiple sequence alignment so that I can show you. Okay, I think it's even no, it cannot be done for now because it will take some time before it finishes. So for the sake of the demonstration, I've already saved this alignment that was run on this same sequence on this particular folder. So if you're going to allow me, I'm going to stop this so that if you run through all this process, you too, you're going to see, or you're going to arrive at the same result that I'll be showing you at the end of the alignment, which, let me open it up. Okay, I say this one is canceled. Yes, I've canceled this. So let me open my folder so that you're going to see this is the alignment that I have saved already, so that I don't just keep us waiting to see when will the alignment be completed, and you'll be able to see it in record time. So once you click on this, multiple sequence alignment that has been conducted using Mzuzu algorithm is going to look like this in our Mega. So sorry, it's coming up because by default it is already in Mega format. So it's going to open it up in this format. So I want to discard these changes to this so that it is going to allow us to have our alignment here back to the same software we are using. So, but please, in case you have finished running your analysis, you can still save it. If you have done the multiple sequence alignments, whenever you are done, you can save it for future reference, so that in case you have a problem with your tree or whatever, and you can still want to go back and start from there, so that you don't have to start all over. So once you click on this, this is how the alignment will look like. You can see this. This is the alignment that was saved. So all it does is to align all these sequences, all the 59 sequences. If you scroll it down, you're going to see them like this. It has aligned them based on the arrangement of the sequence on each of those insulin sequences that I've used. So if I have this, you can see option for save here. So you can save this alignment to the desired folder that you want. I have saved this one already. All that I needed to do now is to construct a phylogenetic tree. After trimming and other things, I can go ahead to construct my phylogenetic tree to study the evolutionary history of these ones. So once you want to do this, it's just for you to still go back again. This is my home page of this Mega. You can see this plan. Remember that this is where we started from. When we wanted to do alignment, we click on align. So if I get back to this home page, what I wanted to do now is to construct a phylogenetic tree. Look at phylogeny here. This is the next icon I want to click in Mega. So if I click on this, it's asking me for the different options that Dr. Wadzani have shown us. If you want to do some of the ones that have to do with character base or distance, you use neighbor joining. If you want to look at maximum likelihood method, these are all different algorithms for constructing phylogenetic tree. So if you're interested in a character base, I go with maximum likelihood. So if you click on maximum likelihood, it's going to now use that same algorithm in order to be able to create or to run this analysis for us. And one interesting thing that I want to show us now is, okay, this is it. I want to construct this using this maximum likelihood, I said. And of course, we have our multiple sequence already. Let me go back to that folder that we've talked about our alignment so that you can quickly see that. Yes, this is my folder where I save some of this information. And this is the alignment we are dealing with in order to construct this phylogenetic tree. So if you click on this, it is going to open up And this is the bootstrapping that he was talking about, because if you want to construct a tree, once it's finished loading, it's going to give you option for the statistical analysis to allow you check what you want to do, how you want to do it, and how you want to run the analysis. Look at this particular interface now. You can see this one now. If my screen is still visible, you're going to see this. Look at it, maximum likelihood tree. This is the statistical method. How do you want to test the reliability of your result? You're going to use this. At times, you're going to see none here. You can see none. It means you are not doing any statistical test. But even the bootstrapping, that gives you different value. You have the standard bootstrap and the adaptive bootstrap. This is what gives you a particular ratio that in Dr. Wadzani's presentation, once we have it up to like 70, it means that branch that has been free is actually reliable, not just by chance. So this is how you test the statistical analysis for this. So, and even for this one, I'm just showing us this one now, because if you're going to run at least from 1,000 bootstrap or replicates, it means this system will run over and over, and it's going to take time. At times, it can take more than 30 minutes to 1 hour still running. In fact, if you have a large sequence, I remember there was one that was running at a point more than because of the nature of maybe the system and environment, more than 6-7 hours we are still bootstrapping, and it's not done. So that is how rigorous at times it can be. So for the sake of this presentation again, this is just to show us how you're going to set up your statistical analysis in form of bootstrapping so that you can continue. There's a one that I've already conducted, the phylogenetic tree again, that we have the bootstrap already, which I have saved so that you're going to see how it looks like, so that we can also see the other aspect of the phylogenetic. Look at this. This is a tree. I'm going to launch this one that I have saved as well, so that we can all see when that bootstrapping is completed using that maximum likelihood, how would the result come up for us to see and begin to interpret and even polish the tree. That is the visualization aspect, and polishing in such a way that it can be more presentable. So I click on that. Let's see how it comes up. Yeah, this is the icon for the tree. Yeah, I don't know whether my screen is still visible and I'm still audible. Can somebody confirm to me, please?
Yes, yes.
Yes, you can.
Thank you. Yeah, so this is, I use the adaptive bootstrapping method. Apart from the standard bootstrapping method, which is actually slow, the adaptive bootstrapping method gives the result that I was able to have from that process. When you allow it to run, after running, you're going to have this as your result. And look at the caption here. The phylogeny was in fact using the maximum likelihood method and just Taylor, Toronto, whatever, blah, blah, blah. It tells you the number of amino acids and everything that was done in this. So all you needed to know, including the citation to this, are already here in the caption. But for the sake of this demonstration, I'm going to hide this caption using this particular one so that we can see the tree very well and for us to be able to talk about it, as well as to do some basic polishing that will make it more presentable. So actually there are different formats of, yeah, let me close this so that you can see this. You can see this. This is 59 sequence. You can see how it's looking. This is a rectangular presentation of this tree. From here, this is the ancestor. You remember Dr. Wadzani in his presentation showed us one tree that has a trunk with so many branches. This is just a reflection of that tree. This is part of the branch which can now become a clade, which means this is the ancestor. Look at the point here with a bootstrap value of 100 is already above 70. So it's telling that from this now, every other thing here, they are like the descendants of this ancestor here. So every other thing becomes a family. This is a branch. We call it a phylogenetic clade, this one now. And these are the different identifiers, which are the ascension numbers for each of those sequences I downloaded from NCBI for representing different insulin from different organisms. So I'm going to show you this. This is one clade. If you now come here again, you're going to see another clade. And look at it, the way I'm scrolling, in order for us to be able to see just 59 sequences. You can imagine when you have more than this. So in order to perfect and polish your presentation, you can actually come here and change the way you are viewing this phylogenetic tree under view. If I click on view, look at what is coming up. And now come here and look at the tree and branch side. If you select this one, I want this one to appear in a cycle. So I will click on cycle so that I'll be able to see this phylogenetic tree changing a format so that from a single view, I can see everything at once, which I want to believe that all of us are seeing this. So from here again, you can go on to say, okay, I want to beautify these three, divide them into the different clades. Look at it. All this one now from this particular branch is representing one clade, and every other here can be descendant. You can go as low as saying this one is another subdivision under this. And look at this angle again. You can have this from here, and you can, if you want, you can turn everything here to be under one branch. And the same thing with this and this. So if I want to broadly do this for the sake of this class, without going into subdivision, I'm going to divide this phylogenetic tree into three clades. One, from this branch, everything that comes start from here down to this place. The same thing with this. I will take this branch this way, from this, and we're going to say we're going to beautify them. And to do this, it allows you to select what we call the node and the taxa. Each of these points where branch occurs is called a node. This one is a node. This one is a node. And this one is a node. So each of them is present what we call node. And what we have here are the taxon. All of these ones are called taxas. The other ones that are here being substituted. So I'm going to select the whole of this from this one now. From, if you can see my cursor, the whole of this from this angle, I'm going to select them and improve the visibility of this so that it can be very beautiful for us and appealing for our presentation and even for publication. So if I click on this, I am going to come down to this sub-3 because I'm dealing with the individual members of this sub-3 now. Once I click on sub-3, I'll go for a draw option and I say I want to format that sub-3 that I've selected. Once you click on it, it's going to show one box telling you that that particular branch has been highlighted. So if you click on that, it is going to allow you to have an interface that can allow you to modify different parts in that particular sub-3 that we have selected, which as our interface. Look at this. This is the node or the sub-3 marker. So if I want to have a marker for each of those taxons that we have there, I can select this, and I'm in love with this particular shape. I can say I want to put this to represent each of the markers that I'm having there. And I can apply a color to represent the branch and even all the members of that branch there. And let me use this first color to select it there. And what do you want to do again? You can improve on the thickness of the line, or you're okay with this. The color of the line, I can apply the same color to this line so that the branches and every other thing there can take a good shape and a better presentation. So once you click on this, thank God, maybe the recording will be available for some of us to follow. I can also come here to change this color. You can even change the font size that you are seeing there. But once I do like this one is going to affect all the taxon markers and their names. I want to change the color. And once I put OK, you're going to see what will happen to the site that I've selected compared to others. Can you see that every other thing that I've selected, which I worked on, is now in this reddish color, where others are in black?
Yes.
Good. So, but look at it. You can also, there's a shape I selected, which is not yet displaying here for the taxon markers. So I can still go back because I want to believe that that thing is still highlighted. I want to put that so that you can see the difference in that knot. The same knot 82 that we are dealing with. Look at it now. Yes. From this, look at this knot or the sub-3 marker. I want it to apply. So I want to click on apply taxon marker. You have to check this box. Apply taxon marker. This particular shape with this color will not be introduced to our taxon marker. I will now put OK. So look at the dot. This is what we have just added so as to polish the tree to take a better look in presentation. So I can say this one is my clade 1. I can come to this side again. Look at this branch. Let me refer to this as another branch from the tree. And I'll click on this. And once you come, the next step I say, you come to under sub-3. And what do? You go to draw options and you click on selected sub-3. And what are you doing? You want to apply another beautification or another polishing to this particular tree. And in order to make it a distinct clade from the first one, I am going to still maintain the same identifier or shape for the taxon marker, but I want to apply a different color this time around so that from a mere view, you will know that yes, you are dealing with a different clade, not the first one. Let me go for this green and I'll click on check this box to apply that effect and even this color to affect the branch lines. All those lines we are seeing, they are branches, and even increase the thickness of our branches from 1 point. I'm using 3 points for now. And finally, you click on display to go to apply the same effect to your taxon names, the knot and everything. Once I check on this, I put OK, and that will now change the second plate to have, look at it, it's already turned to green color with all the other things added. So to save time, I will just apply this. I will consider every other thing from there as another plate. Go to subtree, draw options again, select a subtree, and you repeat the same process. And once you're able to do this, you come up with a more beautiful tree than what it was at the beginning. This is part of the polishing that Dr. Wadzani was talking about. After constructing the phylogenetic tree, you visualize it, polish it in such a way that the presentation can be appealing to others. So in this other color, any color of your choice, you can just pick and maybe let me go with, let me go with this, dark blue or navy blue. The same thing to this, navy blue. Remember to always, if you want that taxon marker to apply, you have to check this box that you want the effect to be represented, and also increasing my branch width so that at least at the end of the day, I'm going to have a beautiful tree. Remember, I'm doing this one, but in case you want to do it in such a way that the names of the identifiers can remain the way they are, no problem. But with this, you're going to have this. This is the phylogenetic tree with the different insulin. And if you want to do it for research purpose or for you to be easily looking at the close relationship, you'll be able to add the identifier of those animals are put right from your sequences. That this one is gorilla, this one is rat, this one is for Homo sapien, this one is for this. And at the end of the day, you have a tree like this, which you have different options of saving it. And if you want to save this, you can see this. You come here, there's an image here, and then you can copy the image, or you can save it as PDF, PNG, SVG, or any format of your choice. You can do this. And you can also save this session using this save icon here. If you click this, you're going to save it. It's going to allow you to save this particular session on this Mega, so that anytime you revisit your software, you can see it's already giving me or taking me to my directory here, and I can save this one as my photo. Anytime I want to rework on this, this one already exists, so it's just a different name that it wants. Or let me update that one, since that one is just a rough tree. So this one will not be my new tree, and anytime I revisit it, I can have it. Thank you very much. This is just a brief demonstration on how to construct a phylogenetic tree from sequences that you have and make it presentable for any of your research work. And I don't think we have the time to begin to explain how the relationship between this and this, how closer they are, and all of those branches. Thank you, sir. Thank you, Dr. Wadzani, for the privilege, and thank you for the organizers too for putting this through.
Thank you very much, Dr. Elkanah, for this insightful and impactful hands-on session. I for one, I'm enjoying it as if you should even continue explaining and explaining. Thank you very much for that, sir. Now I'll move. Okay, if is there any question for this session? If there is any area we need clarity, we can do well to indicate in the chat section, and Dr. Elkanah will quickly provide answers to them. And if there are none, I'll hand over to Dr. Yusuf for him to close.
Okay, thank you very much. Thank you very much, Dr. Elkanah, for this clear demonstration of how to use Mega 12 for phylogenetic analysis. how sequences from organisms can be related to their biological evolutions in terms of their species, and then try to understand relationships and then infer relatedness. So that's very, very insightful. And I think when we get the recording, for those who are interested, you can have a follow-up. We'll also send you other videos that you can follow through, and then you download the Mega software and practice. The best way to learn how to do this is for you to practice. So you continue by training yourself. That's how all of us learned how to use these softwares. But we've had a very good time today, and want to sincerely thank all of you for staying true. I mean, you've paid a very great price, and we hope that this knowledge will be of great benefit to you, and that you also share it with other people, because knowledge that is shared, that's what we're interested in doing, impacting the academia and the industry. And then we synergize to build a better society. God bless every one of you. Ben, do you want to say something?
No. I don't have any other.
Okay, so in closing, I will ask, I saw that there is Dr. Peter Abraham is here. Would you want to give us a closing remark? Then we'll close now.
Hello?
Yeah, Dr. Abraham.
Yes. Good morning, good afternoon, good evening to everyone, depending on your time zone. Want to sincerely, on behalf of the organizers, especially Dr. Yusuf, for putting this very, very important webinar in order to garnish us with essential tools, especially for bioinformatics application, for us to improve our research studies. Thank you so much for this. I think it was so mind-blowing and rewarding session. I want to also appreciate all the facilitators for that hands-on sessions and the presentations. I want to also encourage all the participants to put all that we have learned into practice, because this is especially for bioinformatics and other things. So the more you practice, the more you perfect it. But as you leave it for some time, it will also leave you. So you have to, time to time, go over it again. As we into it, we will be able to make better use of it for our so impactful as well as translational impact in the community. So thank you so much, Dr. Mr. Benjamin. We're grateful. Over. Thank you.
Thank you, Dr. Elkanah. we're sincerely grateful for who has participated. Thank you to the Phyla Biotech team. We hope to, from time to time, be relevant for our academic and industrial advancements and synergy. So somebody was asking whether we could use a Mega 12 for transcriptomics and metagenomics. So depending on what you want to do, you just have to ask questions. And then genomics, you may use another software like Mega. There is Mega Hit, different from Mega 12. And then so many other databases like that with different names, Busco, Metaspades, and all of that. So God bless all of you. And I want to say good night to everyone. Bye.